Hello, everybody. Welcome. Can everybody hear me? Can anybody hear me?
Yes, good morning.
Yes. Good morning. I'm going to give folks just another like 2 minutes or so. We still have kind of people filtering in, and also for me to figure out how to find my notes. All right. Can everybody see the slide that I have up? Beautiful. Great. Okay. Give like one more minute to get the rest of folks in here. Okay. So let's do this. We still have some folks joining us, but I'll go ahead and start with the introductions. So thank you all for coming at bright and early at 9 o'clock in the morning to the Atlantic Coast Freshwater Mollusk Symposium. This meeting kind of got sparked from the idea that we used to have a Atlantic Slope meeting for a while pre-COVID. We liked the collaborative nature of it, but then it kind of faded away, as did a lot of things after COVID. So we wanted to get it going again, bigger, better than ever. So we decided to invite everybody up the East Coast and see what happens. So welcome and thank you for joining. The big goal for this meeting today, I was just hoping to strengthen our collaborative networks. We've had some turnover in the field, so get people and some students to connect with others, see what everybody else is doing, and just chat about some problems if we can get some solutions to them going. See where it goes. Let's see, got everybody in? So the schedule for today, we're going to keep it kind of loose because there was quite a few people that needed to be in the field, so they'll be popping in and out as we go. But this morning, I'll start with a quick welcome, tell you about who I am if you don't know me. Then we'll do introductions from everybody in the group that can do an introduction, anybody that wants to. Again, the point was to like make connections here. So that'll be, I sent out an email, most of y'all got it. So a couple words about who you are, what you're working with, who you're working for, what kind of projects you're doing, anything like that is really helpful. The meeting today will be recorded, so we can put it up later for anybody that didn't make it to the meeting. So that's a heads up. I'll probably say that again in a couple of minutes just so everybody's aware of it once everybody has settled into the meeting. That should go, I think, until about noon or so, and then we'll take a lunch break because we're all going to need one by then for sure. And then we had some volunteers for this afternoon to do longer presentations. We had a couple of groups that wanted to announce what they're doing. We had a couple of students that wanted to talk about their work. So I'm excited for that. We'll have a chat, wrap up, and see if we want to do this meeting again, how everybody feels how it went. So again, thank you for joining. If you have to pop in and out, totally cool. Just keep that mic muted if you don't mind, and then I'll admit you back in as you come in. Okay, so a couple notes as we roll through the meeting. Keep an eye on that chat. I'm going to ask some folks if you can do it to drop your information in the chat. Hopefully I can put that all together into a document later, so you don't have to worry about taking notes too much throughout the meeting. To break up the monotony of 1,000 introductions, because I think we're looking at about 60 people so far with us, which is a better turnout than I expected. So thank you all for coming again. Yeah, so to break up the monotony of 60 different introductions, I'm going to have a couple discussion questions, just basic level, so we can take a break, chat, take a bathroom break, do whatever you got to do, then come back on it. I'm also going to put a contact sheet in there so you can add whatever information you want to the contact sheet. That saves me having to do it manually later, and everybody can have access to it if you want to share your contact. No pressure if you don't. In fact, I'll just go ahead and drop that contact sheet in there right now. Make sure I know how to use the chat. Oops. Get big. All right, chat. This should go to everybody. All right. Hopefully you have that link now. That's a Google Doc. I'll send it out again later. I know there were some issues with Google compatibility with emails, so if that doesn't work now, I'll get it to you later. And if you could update whatever you want to share in there, that way we all have a resource so we can keep in touch after this meeting. Good. What else do I have? 1,000 tabs open. All right. Okay, so let's start. So there's two ways you can share in this first part. If you are comfortable sharing your slides or talking for a second, I think that would be the most interactive way. So I would love for anybody who wants to do that to go ahead and raise your little virtual hand on there. There should be a button. And then I'll just go through the list and call on people as we go, whatever order they show up. There was a couple of folks that told me they have time constraints, so I'll prioritize them around their time. And if you have any that you haven't mentioned to me yet, you can drop me a message in the chat and I'll check on that and try to make sure we can stay respectful of everybody's time. If you are traveling or don't want to participate in the audio, let me drop in an example of what you could put in the chat and you can introduce yourself this way. There you go. Something like that. Then we can have a record. Hopefully I'll have the chat printout at the end of this too, so everybody can have access to that. Any questions so far? Everybody feel good? Super. Okay. Perfect. Let me make some notes here about folks. Perfect. Okay, so for those of you that don't know me, my name is Loretta Lutakis. I am a conservation aquaculturist at North Carolina State University. That's me up front with two of my former lab mates. In the back, I am also a PhD student currently under Dr. Greg Cope. What I do here is I study in vitro propagation of freshwater mussels. I'm really focusing on the development and doing a serum trial of them. So one of the things I'm doing, you can see in that picture, is thin sectioning Glaucidia and then making a full developmental timeline of them and how different sera affects that development. I also work with the magnificent Bramshorn, so doing a similar thing with them. We've done a little bit of development work, but otherwise a lot of propagation work. We work really closely with the Wildlife Commission here and the Marion Conservation Aquaculture Center. They're doing a similar thing to that. So you'll probably, if Sierra's on today, you'll hear some of that too. If you're here, you probably got my 1,000 emails. So that's my email address and my contact information. But there it is again, in case you need it. Cool beans. Thank you to everybody that's dropping their information into that chat. I really appreciate it. So we'll go ahead and start to introductions. I was going to hand it off to my lab first as a good trial run to make sure everything is working here, and then I'll start going through everybody that is interested in sharing. So give me a second to swap these screens, and then you can hear about my lab. Do this. Alrighty. I think y'all should be able to see a slide that says Yates Aquatic Conservation Center now. And I seem to have lost my Zoom, so we'll find that again in a second. There it is. Okay. All right. That slide look good to everybody? Beautiful. Here is Chris to talk about that then.
Thanks, Loretta. I'm going to just jump in and give a quick 2 or 3 minute introduction to what we do here at the Yates Aquatic Conservation Center at NC State University. This will be won't be new to some of you, but will hopefully to others that aren't familiar with us. We're part of the Department of Applied Ecology at NC State. This is our facility. We are about 4,400 square feet of indoor and outdoor lab space. We run on 4 full-time staff. That's 3 biologists and 1 technician, and then plus 3 student interns that we have from NC State. This is our water supply. We sit on a historic mill pond and within a Wake County park. It's about a 20-acre pond, and we pump water in for the culture of freshwater mussels, fish, and snails. We originated, were established in 2023 via illegal settlements over the completion of Highway Interstate 540 around Raleigh. This interstate passed through the Swift Creek watershed within the Neuse River Basin, and the Swift Creek watershed is home to 5 federally listed aquatic species, 3 mussels, also the endangered fish, the Carolina mad tom, which we work with, and a salamander, the Neuse River water dog, which is a threatened species, which we don't work with yet. This is our mission. We see ourselves as helping to conserve North Carolina's imperiled freshwater species through propagation. Again, that's mussels, snails, fish. Research, we do a variety of research. Loretta will be talking about her in vitro research. We've had various grad students come through. We're working on mussel rearing, fish rearing. The grad students have done things like mussel health, had a genetics project, and beyond. We see education as being at a university. This is a major part of what we do. Bringing in interns, not only are they helping us, but we are hoping to help train them to be, start off the next batch of biologists on their career path. And then finally, outreach. We run tours to the public, to school groups, about once a week. We have an online presence, just trying to tell people about the beauty of freshwater mussels and the imperiled aquatic fauna that we work with. Here's some of the species that we work with. These are 10 species over the last 2 years. We do about 6 or 7 each year. Of course, some of the ones we focus on are those most affected by by the road crossing, those listed species, but we also work with other state listed species on the Atlantic slope, from anodontines, some amblyomines, short-term brooders, and also a variety of lampsalines as well. And just a real quick view of what the inside of our hatchery looks like. This is our fish holding area. When we don't use fish, this is what our in vitro lab looks like. Our mussel rearing, this is a part of it. As we're growing, the areas of the lab where we're growing mussels are expanding. We're basically using a pulse flow through system and just giving them more space and more water as they grow. And then finally, our final grow out, we have a mixture of an outdoor kind of a screened in porch area where we're doing flow through grow out. And we're still doing some remote grow out in Lake Myra because it's another reservoir, I guess, east of here a little bit. That's been so productive for us. And with that, I will leave you with a picture of one of our mussels from down here in North Carolina, the yellow lamp mussel from the Deep River, North Carolina. And I will turn it back over to Loretta. Thanks.
All right. Thank you very much, Chris. Okay, I think we have most people in that are going to be in the meeting, I would think, right now. So hello again to everybody. Anybody that I haven't already said hi to. Hi, I'm Loretta Lutakis. I'm at North Carolina State University, and I'm hosting your meeting today. Quick shout out that the meeting is going to be recorded if you missed that memo earlier, so it can be shared later. And also, at the very beginning of the chat, there's a contact sheet. So if you don't mind sharing your contact so everybody can keep in touch if they want to, drop that in that contact sheet today would be super helpful. Okay, so I think we'll move on to other introductions. For anybody who wants to do a quick audio introduction, share some slides would be fantastic. I'm going to start with the folks that had time constraints. Art Bogan, are you on here this morning?
Yes, good morning.
Good morning. Thank you for joining. Are you in a good place to go ahead and share? Say hi.
Yeah, yes, just a moment. I'm Art Bogan. I'm research curator of mollusk here at the North Carolina Museum of Natural Sciences. And I wanted to talk to you this morning about excitement in taxonomy. I realize that most of you just need a name and are upset that every time you turn around, the names are changing. You have two options. One, Freshwater Mussel Conservation Society on their website since 1919 has been putting up every 2 years a revised, updated list of the freshwater bivalves of the United States and Canada. And we're looking at adding Mexico to that list. We're also looking to add fingernail clams, Spiridae. Right now, there are 4 appendices, the 2019, 2020, 2021, 2023, 2025 up on the website. Gives you an idea of how things are changing, what names have changed, and it's free to download. It's on the front page of the website. Yes, things keep changing, and it's a moving target. The alternative to this, in 2017, James Williams and friends had published a paper kind of summarizing where we were in 2017 after the last edition of Turgeon et al. Common Scientific Names of Mollusks in North America. We're in the process, John Harris is leading the crew to develop a list of up-to-date freshwater bivalve names. Like everything else, once you start writing, it's out of print, out of date. We have in press a revised, updated list of spirids of North America, United States, Canada, and Mexico. And it hopefully will be added to the list that we're producing. If you want to be more up to date, the Mollusca base online database has about 38 international editors and is constantly being revised and updated as new material comes out. If you're having trouble finding a name, it's an excellent way, and it's free. And if you have questions about it, the editors are usually very, very prompt and helpful in responding to any questions. It's a moving target, and just because it's the names you used in graduate school, it's not what it is today. And if some of your, how to put this delicately, more mature, you go, I'm tired of changing names. As science moves on, things are going to happen. Names are going to change, especially as phylogenomics increases and the techniques that are improving. We're getting more information and hopefully a more refined understanding of the relationships in the order of Unionida. If you have questions, send me an email. I'll try to answer them. And if you have a problem or if something's wrong, please let us know. John Pfeiffer at Smithsonian is the new chair of the Common and Scientific Names Committee for bivalves at FMCS, and we're always happy to talk to people. With that out of the way, we can get on to the fun part of the morning. Thank you. If you have questions, let me know.
Thank you so much, Art. All right. Anybody have quick questions for Art? Cool beans. Let's see. Apologies in advance if I say your name wrong. I'm probably going to butcher most of them, but I promise I'm trying my hardest. Let us see. Time constraints. Philip Mathias, are you here and did you want to share? I saw that you had a bit of a time constraint on there too.
Yeah, morning.
Good morning.
I work for the Fish and Wildlife Service, U.S. Fish and Wildlife Service, and at the New York Ecological Field Services office. I'm just past my probationary period, so been a bit of a rough year. So I don't have too many like project updates. We've been fairly restricted. Um, However, I am the national lead for the dwarf wedge and brook floater and green floater. Right now, everyone keeps asking, but there's no movement on green floater. Everything's just kind of stalled. But we are starting our 5-year review of dwarf wedge. We have our first coordination meeting internally next week. So I believe our listing coordinator reached out to many of the states, but if you need a reminder, please send us any kind of dwarf wedge updates you might have if you're a state lead. And yeah, we've looked at the brook floater a little bit more. And I'm also a technical lead for our at-risk mussel species, which has a lot of the Atlantic slope species on it, like yellow lamp mussel, tidewater mucket, um, brook floater as well still. And then kind of one thing we're trying to get a nationwide system out. It's called the Program for Unionted Long-Term Standardized Evaluations. So our Virginia field office mussel biologist is developing kind of sampling techniques for non-lethal hemolymph sampling to kind of get a baseline on mussel health. So we're trying to get at least one unit of these sampling units in each state. So maybe hearing more about that coming up. So I think that's all.
Thank you very much for sharing. Allison just messaged me with a great idea. So as we go through, I'll put a couple of folks like on deck in the chat. So if that works out for you, you can share. If I call your name and you don't want to share, just go ahead and pass. No big deal. But then we can keep it, I think, flowing a little easier. So Allison, actually, if you don't mind going next while I start getting that prepped, if now is a good time for you, that would be great. And then we'll follow up with Chrissy if that's okay for you guys.
Sure thing. I probably deserve that.
You made it an easy call next. I was like, oh, great, a name. Let's do it.
So my name is Allison Roy. I'm with the U.S. Geological Survey. I'm part of the Cooperative Fish and Wildlife Research Unit program in Massachusetts. I'm at the University of Massachusetts Amherst and have an adjunct appointment there. I wanted to highlight, so I've had I've been working here for 13 years, and I've had several graduate students and a whole slew of undergraduate students doing research with me in that time, heavily in collaboration with the state agencies. And I wanted to highlight a couple of things. So I'm going to share my screen. First, in the interest of recency, I wanted to mention a couple of publications that came out of our lab in the last 6 months. One about prioritizing, so both are based on species distribution models from the Northeast states. And we use them to inform understanding of what are the drivers of freshwater mussel population, so all of the species that we have in the Northeast, and approaches for conservation. And then we also did some modeling with projected climate change to see which watersheds were most likely to change and be vulnerable to climate change. And this was based on both mussels and fishes. So it was neat that the mussel piece could include host fishes. So just a highlight of those two papers, both are online and available freely, open access. And then the other thing I wanted to highlight was one of our working groups. So, oops, get rid of the animation. So the Brook Floater Working Group, many of you on the call are part of this. For those of you who are not and interested in Brook Floater, I just want to give you a brief description of what this is. So this is a group of folks working on this species who are interested in conservation of this species throughout its range, which really covers almost the entire Atlantic coast. And we have representatives from a lot of different organizations. People have been changing through time, and so I appreciate those of you who've reached out and said, hey, add this person to the list. We meet every other month, and we share research, but we also talk about the problems we're encountering, the opportunities that we have. For example, we had a meeting talking about propagation and what challenges we're facing and what future opportunities we have. So real mix of topics throughout the year, really to keep the conversation going and work toward widespread conservation. So this was developed based on a competitive state wildlife grant that was awarded to the states that are listed on the bottom of that screen. We had a first one in 2016 to 2021, and you can see some of the things that we accomplished there, notably our survey methods that have been used widely, and a lot of work on propagation and planning for restoration. And then we successfully received a second CSWIG that we're in the last year of, and these are the funded states for that, also working with universities to do a lot of the research. And our project components are similar. We're taking steps forward for modeling based on a ton of surveys that have happened in the same way throughout the entire range, which is really cool. And also lots of other pieces. So I encourage you to check out the website that's listed on the bottom if you want to learn more about what we've been doing, and reach out to me if you want to be added to the working group email list. Thanks.
Thank you so much.
So I guess I'm following Allison. I'm Christina Murphy. Most people call me Chrissy. I'm also with the USGS Cooperative Research Units, but I am at the Maine Cooperative Fish and Wildlife Research Unit, which is at the University of Maine in Orono. And I'm also a faculty member in the Wildlife Fisheries and Conservation Biology department here. And I will talk a little bit about the yellow lamp mussel working group. So similar to the Brook Floater working group, this is sharing, right? I still just see myself gigantic. Okay, I'm sure this is fine. The yellow lamp mussel working group, you can go to the website and I'll put it in the chat. Similarly, a leadership team comprised of university and federal and state partners. We have resources listed on the website of different presentations. We meet bi-monthly. If anyone is really excited hearing about yellow lamp mussel and bi-monthly meetings, our next meeting is on Friday. So you can contact me for the link and information. And we are an open group. We maintain a list that you also can come in and edit. So we have 67 people right now that typically attend the meetings and participate. And you'll hear more from the graduate students that are leading a lot of the research in the afternoon. So with that, I think I am done. Unless I've forgotten anything, and then Allison, Jill, and Stephanie, I think, are all here and can jump in.
Sounds great. Thank you so much. I think as we go through, if anybody has questions, feel free to pop in during this transition time or drop them in the chat. That's good too. And thanks for putting that in there too. All right, next up, let's do Pete Hazelton, please. And then we'll follow that with Amber Olson.
Thanks. Thanks, Loretta. My name's Pete Hazelton. I'm an assistant professor at the University of Georgia. I know a number of folks here. My lab has been involved in a number of different studies recently. I'm a co-facilitator of the Brook Floater Working Group with Allison, and so this is a good time for me to go on. But my lab works a lot on survey methodology, population estimation, and then, but we also do a lot of work in ecotoxicology and a growing interest in mussel health. So we're down here in Athens, Georgia. We have a pretty significant presence right now in the Savannah Basin, working with South Carolina DNR, and then are doing some work in the Altamaha as well. And I should also mention we do a significant amount of life history and host work as well. So if you have any interests down in the lower Atlantic slope, would love to talk to you. And I'm going to be hopping in and out throughout the day. And I'd love to learn more about what folks are doing in the area. Thank you.
Thank you so much. Amber, you good to go?
Yeah, can you guys hear me?
Yep, sounds good.
Awesome. My name's Amber Olson, and I work with the North Carolina Wildlife Resources Commission. And just wanted to give a few updates, just kind of about what we do, at least in my region. So I work with two other folks in the eastern region of the state, and our primary focus is in the Tar and Neuse River basins. We've got a few different listed species there that we really focus heavily on. So Elliptio-lanceolata. We've got Fuscania masoni. We've got the Tar River spiny mussel, Parvispinus steinstonsana. So we work a lot with these species. And this past year, we've done a lot of stocking. So for Fuscania masoni, the Atlantic pigtoe, we've stocked over 2,000 mussels in the Neuse River Basin. We just recently started to stock Strophitis undulatus, so the creeper. We stocked about 870 of those in Swift Creek. And we also work with the dwarf wedge mussel. So definitely going to be getting into contact with you, Philip, because we haven't been having great luck with that mussel. This past year, we found one female and I think 3 males, and we weren't able to. I think, Loretta, you might be able to speak on this. None of the progeny survived, right? For this,
I'd have to double check with Chris, but if they did, it was just a very small handful. I'm not sure that any made it this year.
So that's a species that has been struggling a lot that we work with. And then with the Tar River spiny mussel, that's some species that Mike Walter is going to be getting more an in-depth presentation later on today. But that's a species that has really declined in the Neuse River Basin. So we really only see it in the Tar. And we've been doing a lot of really cool work with it. So we have a long-term monitoring station where we have released a lot of these mussels, and we go out and do really like in-depth quantitative survey roughly every 2 years. So this past year, we just did that, and we found potential recruit, which is really cool. And a few other wild tar spinies. So Mike will talk more in depth about that later. And then in this upcoming year, we're going to be focusing on developing eDNA primers, or I guess those have already been developed. We're just really like making sure that those work, ground truthing them. And so hopefully we'll be able to find some more wild populations of the species. We're also going to be doing a silo project where we take some of the young year tar spinies, put them into silos. Hopefully most of y'all have seen similar projects just to kind of get an idea on how water quality could potentially be affecting their survival. We are going to be partnering with NC State, doing this long-term 10-year water quality monitoring project that we're going to try to pair that silo experiment with. And then another really cool thing that we're starting to work on, let me see if I can share my screen, is doing micro CT scanning on tar spiny mussel shells so that we don't have to physically section them in order to age them. Let me see if it will let me. Share my screen.
Should be
So
along
that
the
is something.
bottom there. Oh, yeah, there you go.
Again, this is with NC State. They've got a really, really cool micro CT scanner. And I don't know if you can kind of see it. We've got what looks like potential annuli here on the scan. So the next step would be to validate these annuli with known age shells with like physical sectioning to make sure that that's matching up. But that could be a really cool method to use in the future just so we don't have to sacrifice rare and valuable shells. And that's something that my coworker, Michael Fisk, has really like developed. And yeah, just a bunch of random updates with things people might find interesting. So if anyone has an interest in those species, reach out to me, Mike Walter, or Michael Fisk. All of our contacts, I think, should be on that sheet.
Thank you so much. Those look wildly cool. That's exciting. How about Erica Beeson? You all right to go next?
Yeah.
Good. After John, I don't have any more volunteers right now. So if you are volunteering to share it, if you could drop your name towards me, that would be really helpful. That'd be great. Thanks. Go ahead, Erica.
Yeah. Thanks, Loretta. Hi, everyone. I'm Erica Beeson. Ignore my voice. I am getting over being sick. Just a few updates from South Carolina. First off, this is our first successful year producing and maintaining brook floaters. So we've got some brook floater babies, and those guys are going to be used in a silo study with UGA as part of the competitive SWIG grant. So very exciting, very cute little babies. We work mostly with propagation of Platanias decorata, which is a new name. Thanks, FMCS Taxonomic Committee. And then just kind of the biggest update that I wanted to share is that we are finally kind of wrapping up our conservation benefit agreement for the state, specifically for freshwater mussels. And that is what used to be the safe harbor. So they combined the safe harbor and candidate species, and it's now called the Conservation Benefit Agreement. This has been years in the working, and it's finally getting to where we're gearing up to submit that to the service this year. So if anybody has any questions about the Conservation Benefit Agreement or kind of what the framework looks like or kind of the process about that, please email me. I'm happy to kind of tell you what our steps were to kind of get to that point. It was very nice that North Carolina already had one established. So thanks, North Carolina. But then the other thing, just kind of update wise, I'm going to share my screen here for a second. Excuse me, we have started a new survey type. Can y'all see that? Yeah, we've started a new survey type. So again, we work mostly with Carolina Hill Splitter. That's our one federally endangered animal in this state. And you could see that we were not finding any animals, literally like finding one. So in 2018, when the program was started, they designed standard catch per unit effort surveys so that they had standardized sampling across watersheds and populations so that they can compare year to year, and especially in the 5-year review, how the populations were doing. And this past year, I was like, we've got to find some animals. So we started a new survey type that we're calling the sweeping surveys. And you can see just how many more animals we have turned up in these sweeping surveys. So we still have standardized sampling procedures so that we're able to compare across populations and watersheds. But you can see that in 2024, we found one animal across all of the catch per unit effort surveys. And this past year in 2025, we found 47 animals in those sweeping surveys. So we went from finding one to 47. So if you have any questions about kind of that, the true like needle in a haystack animals, I mean, you can find thousands of mussels and only one heel splitter. So just how we've been able to turn up these animals, feel free to shoot me an email. But that's all I've got the voice for. So I'll stop now and excited to hear what everybody else has. I'll be popping on throughout the day. So yep.
Thank you, and I hope you feel better.
Thank you so much.
Yeah. All right. Next, if we could have John Pfeiffer, please, and then we'll do Heather Evans, Michael Perkins, and Alex Crawford.
Hey, thanks. Can you guys hear me okay? Great. I'm gonna share my screen. Y'all can see this? Great. Thank you. My name is John Pfeiffer. I'm a research zoologist at the Smithsonian. I'm also the curator of bivalves. So I just wanted to kind of update folks on some efforts we've been undertaking at the Smithsonian, aimed to help conservation biologists and researchers better understand freshwater mussel biodiversity. The first is this interactive web app that I developed that like collates natural history collections data from 45 different natural history museums and kind of puts it in a centralized location that allows folks to kind of easily navigate freshwater mussel occurrence data. So you can go to this web page and search by whatever species you want, and it'll pull up a map, and then you can zoom in to like look at individual freshwater mussel records. It'll tell you what museum it's from, what river, date if it's available, the stream order that it was found in. You can click on any one of these dots to explore these occurrences. It also produces like a Huck 6-level map for every species, again, based exclusively on natural history collections data. A histogram about how the species has been collected over time, like there was a ton of it collected in 1992. Information about its ecological preferences based on natural history occurrence data. So like this species is more commonly found in third and fourth order streams than it is in seventh order streams. Individual species is found from 222 georeferenced natural history records from 37 different Huck 8s. This is the Huck 8 area of occupancy. Just kind of some summer statistics based on collections data. And you can download all of these occurrences for this species using this button. By watershed. Button, and then select your Huck 6. And again, pull up a map and you can click individual records to learn more about those occurrences. Here's like based on those occurrences, a species list of all the species that have been found in that Huck 6 over time. Again, here's like, Elliptiocampionata on this end, and then some very uncommon species on the other side. Yeah, hopefully you all find that useful. And if you have questions or concerns or improvements, please don't hesitate to reach out. In that we have just completed like a mass digitization effort of our freshwater mussel collection. So now essentially every dry freshwater mussel record in the Smithsonian has been imaged. So you can go to our search portal and type in a name, species, oops, dates right now. Plus, it'll pull up more information as well as thumbnails of the specimens from that record. And so we take 3 images. specimen, a door or a external view, an internal view with the insides of the labels because there's often useful information written on there, and then a dorsal view. And these are really high resolution images, so you can zoom in. It's got a scale bar, color bar, all the label data. About a record or want to see the specimens. These are some really great images to help make our collections more digitally accessible. We've also been creating 3D models for a bunch of different freshwater mussel species. So we have over like 500 species that have a 3D model now. So you can click the models. These are all Smithsonian specimens, and you can and zoom in. Models here. And so like people have been using these to like 3D print them for like training and education and outreach. Where you a very in the hands of a kid on a boat. You could create these 3D models and use these as kind of education and outreach opportunities of sensitive material. Is the Smithsonian has lots of different fellowships and internships. So if you have students that are interested in a freshwater mussel project, they might be able to apply for some of these fellowships and undertake a project at the Smithsonian on how natural history objects could help inform conservation. So please don't, and I'm happy to answer any questions. Thanks.
Loretta, can I ask a quick question?
Yep.
John, I missed how you got from the mussel records mapping to the 3D page. Was that a different link?
Yeah. Throw it in the chat right now. It's
okay, yeah. Thank you.
3d.si.edu. And then at that page, you'll probably just want to go to explore and type in Union a day,
yeah,
or something like that.
Thank you.
I can actually put a more specific record. Models are so realistic, it's crazy. Obviously, the weight feels different, but if you use like a Mamaki printer, the color is beautiful and it will fool even experienced malacologists if you were not to pick them up.
Thank you so much. I think we had another question, if that answered all of your questions. Maddie?
Yeah, John, that muscle mapper tool is so cool. We use it all the time. I was wondering if there might at some point be a capability to have an actual location search bar of some kind so that you're not just kind of zooming in on the area that you think you might be looking at.
Sorry. I'm always open to suggestions, and if I can make them happen. So just want to make sure I understand.
Yeah, just so like similar to like a Google Earth kind of thing where you can either plug in coordinates or an address or something like that. Because otherwise, like I'm still kind of getting used to the geography here. So I got a lot of cross-referencing of trying to zoom into where I think I might need to be.
We could make that happen. Yeah, the closest solution right now would be to click the watershed and then zoom in from there.
Yeah.
Well, yeah, I think I'll just leave it there for now. But yeah.
Well, no problem. Thank you.
Let me try that again. We had one more question pop up in the chat for you. They were Mike Walter was wondering if the scanning was done in the field.
that's done in the museum, it's done like just camera, like taking hundreds of images from all the different angles and then stitching those images together. So it could technically be done in the field, but we have all the specimens that we're trying to model are in the museum, and it's just a little bit easier. But you could technically create one of these 3D models from a camera in the field.
Super. Thank you so much. All right. If everybody's good, we will carry on. I think we'll do these next three, Heather Evans, Michael Perkins, Alex Crawford, and then just take we'll be going at an hour or so maybe a quick 5-minute bathroom break. If everybody is good with that. All right, Heather.
Hey, everybody. I'm Heather Evans. I'm the Conservation Genetics Manager for the North Carolina Wildlife Resources Commission. So I thought I would just give a quick update on some of the tools that we've developed this last year. So we had a graduate student, Edie Nissen, who Greg Cope and I co-advised, publish it for her dissertation, a parentage-based tagging panel for tar river spiny mussel and yellow lance. So those panels are meant to help us monitor stocking efforts. They should give us an idea of hatchery contribution. And also we designed them such that they would allow us to track population structure as well. So those have been published in her dissertation, and we're working on getting them published in a journal right now. And then we're working on developing 2 more PBT panels, one for dwarf wedge mussel and one for Atlantic pigtoe. So I can share, let me see if I can figure this out. Here we go. So I'll have to click share. Okay, so hopefully you guys can see this. So this is when we say Scott's, this was a presentation I gave earlier. I just pulled up a few slides from. So Scott is Scott Myers, who is at Georgia Southern and Jamie Roberts' lab, and he did a population genetics analysis for us using thousands of SNPs. And so what we did was pull from his SNP panel to reduce that number to something that we could use in what we call a GT-seq panel, which is basically like a multiplex PCR, but it reduces the amount of SNPs such that we can do this high throughput monitoring. And you can see that using that reduced number of SNPs, we're able to still get the same kind of population structure. So that'll be useful for monitoring, we hope, in future. And my mouse, there we go, was not working. And now I can't advance anything. Oh, I don't know how I did that. Okay, well, we're going to keep going. So there's a similar structure plot for the Atlantic pigtoe. And those are currently in development. And then the other thing that we did this year was Amber mentioned this, develop an eDNA assay for tar river spiny mussel. So these are all of the species that we tested against this assay, everything that we knew was co-occurring for tar river spiny mussel. What it amplifies is both tar river and James River spiny mussel. So we think that it can be used for both, although caveat is that we've only done the lab validation and the field validation for tar river spiny mussel at this point, but it is amplifying James River spiny mussel, but we haven't tested that in the field. We did test tar river spiny mussel in the field, both at negative sites, which we didn't amplify anything, and at a positive location. This was multiple distances downstream from a known stocked location, and we can get detection as far away as 300 meters downstream from that stocked site. We didn't test further out than that. We think this is a pretty nice linear regression we don't see with fish because they move around. So we think if this continues with that same linear model, we can probably get detection around 700 meters downstream of a mussel bed that has a certain biomass. So we're going to do some further tests this year, looking again, how far downstream from that bed can we get detection. We're also going to do some cage trials with known biomass and fewer mussels and look at how far downstream we're getting detection from that. And then, we're going to be using it to do broad geographic surveys and hopefully find some new populations. So that is what we have been up to. And if I can get my mouse to work again, I'll stop sharing. There we go.
Thank you so much.
Yep.
Right on. Next up, we've got Perkins.
Good morning.
If now is a good time for you.
Yeah,
Great.
can y'all see me and hear me?
Yep.
Good morning, everybody. I was just going to share a quick update. I think I know most of y'all, but for folks I don't know, I'm the Foothills Regional Research Coordinator. I work for the Resource Commission in North Carolina, and we've been working a lot on just over the last year dealing with the destruction from Hurricane Helene and the debris removal aftermath. Both were really consequential for the mussel populations in the western part of the state. And I don't have any like data to, show to anyone about it yet, but we've been working a lot on that. I did just want to share really briefly two pieces of research that we had published last year that I was a part of. I'm trying to share my screen, but I don't see the thing for some reason. So
Maybe along the bottom, although it seems like a couple of folks have had trouble finding
yeah,
it.
it's not coming up for some reason. That doesn't matter. The one of the big things that we finished last year was a description of a new endemic mussel in North Carolina, which is Lagodona obscura. It was a 9-year or so project that I was the lead on, and we worked, lots of people in North Carolina and elsewhere worked on it. Several of y'all probably heard about it, but if you haven't and you're interested in the paper, it's in Zookeys, it's open source. And the most interesting thing about this is that it's like this insanely narrow endemic. Right now, we only have a currently extant known population of about 8 kilometers in one contiguous stream reach. So this is something that I think we'll be working on a lot more. And yeah, who knows what's going to happen with this thing in the future. Being that rare is not good. Another piece of research, this was in a journal called Science of the Total Environment, working on dams and hydrology. This was a partnership with Virginia Tech, and this paper was isolating the compounding effects of dam hydropeaking and climate change on freshwater mussel recruitment, specifically on James spiny mussel, but I think some of these findings are applicable to other species, at least like broadly in the Blue Ridge or Blue Ridge adjacent streams. But yeah, that was looking at how, I mean, without being too like talking too much crap about hydro dams, pretty much looking at how hydro releases are destroying mussel recruitment. So if that's a topic that's interesting to you, I'd highly suggest you check the paper out, or you can email me and I can point you to it. All those papers are also on ResearchGate. So yeah, that's all I got.
Will you pop a link to that hydro peaking paper in the chat, Michael?
Yes, I will.
Thank you.
Thank you. Thank you. If that is all the questions, we'll go on to Alex Crawford, please.
Hey, can everyone hear me?
Yeah.
Yeah, perfect. Hi, everyone. Feeling a little out of place here. I didn't realize that much work was going on in North Carolina. I'm in New Brunswick, Canada, so a little bit out of the way to you guys. I'm a master's student at the University of New Brunswick here, working on mussel ecology. That's my project, specifically on Ian Grand Lake. That's our largest lake in the province. I can share my screen here, I think. Did So can I make that larger for anyone? There. No. Aha. Hopefully that works if you guys can see those pictures. Anyway, so I've been doing a 2-year study on essentially just a population survey, as well as trying to find yellow lamp mussel, and narrow down specific sightings in the province, because the last time it was done was in 2013, and we're needing an update on that to see where they are and how they're doing. So I'm still processing data on if we actually found them or not. But overall, I was going through the different 8 species that we have in Grand Lake and doing a mussel methodology survey evaluation, I guess, for locals to use. So, but I'll give a longer presentation on that later. I think that's all I'd have for the quick update there. Just excited to hear what everyone else is up to as a student.
Thank you so much, and thanks for joining us. I think you probably win the most northern here. All right, so I'm thinking let's take maybe a 5-minute break real quick. I'm gonna drop a topic for discussion in the chat real quick for anybody who's gonna stay, and I'll leave the line open if you guys want to chat for just a couple minutes. It's just feel free to answer if you want to. If not, that's totally cool. It was just kind of a thought to see if there's any way that anybody on this phone can help everybody else out. So there you go. What's a challenge you're currently facing, and collaboration could help with it. So if anybody, students especially, if you're looking for information and you haven't found it yet on this call, feel free to throw a shout out in there. And let's see, we'll see you all back here at like 10:06-ish. Thank you. Alrighty, welcome back everybody, and thank you for more volunteers. We're getting you all on a list. If it's all right, I'll still keep announcing like 3 at a time because occasionally people have to jump out, so I'm gonna move them around. If that's okay with you, 3, 4, a handful at a time. Great. So who did I say? I think Langston. Did I say you were next? Yeah. Let's do Langston if you're back.
Awesome.
Let me stop sharing my screen too. There we go.
All right. Hey, y'all. I'm Langston Rimmer. Sorry if I'm a little coffee. I'm under the weather today, but I just wanted to give a few updates. I'm a biologist in the central region with the North Carolina Wildlife Commission, so I cover the Cape Fear, Yadkin PD, Lumber, and White Oak River basins on the Atlantic slope. And so I just started this position over the summer, so really haven't done a whole lot, but had a few updates. We did some surveys for Atlantic pigtoe in the Cape Fear River Basin. We haven't seen them there in the basin in general since 2017, I believe. So we're really hurting to find an individual so we can start some, get some propagation going with our partners. But that's one thing we did, and we did not find any this year. We put in about 70 person hours, so not a huge survey effort, but we focused on New Hope Creek in Duke Forest, which is where they were most recently collected. Mike Perkins already talked about the description that they worked on with the Solstice Creek mussel, but that's something we're doing right now, is working on host fish, understanding the host fish with those through research. So we collected a bunch of potential host fish to do some trials. So no results from that yet, but hopefully we'll have those next time we have this meeting. And then we also did some relocation surveys associated with several dam removals occurring in the deep river of the Cape Fear River Basin, and took advantage of those broad surveys to collect some individuals for propagation. So we've been propagating several mussels with NC State, brook floater, yellow lamp mussel, Savannah lilliput, and eastern creek shell. So looking forward in the next year for our region, we're going to be focusing a lot on hopefully finding some pigtoe in the Cape Fear, looking more into research and monitoring surveys associated with the Solstice Creek mussel, and continue to do some of our long-term monitoring efforts that we have in the PD River, as well as the kind of evolutionarily unique habitat of Lake Waccamaw. So nice to be here with everyone, and look forward to hearing about everyone else's projects. Thanks.
Thanks, Langston. I think I'm up. Should I go ahead, Loretta?
That sounds great. And then we'll do, let's see, who do we got next? Justin Lewandowski, please, after Aaron.
Awesome. Well, Loretta, thank you so much for organizing us. And it's so nice to see so many folks that love and care about mussels. I'm Erin McCombs, Southeast Conservation Director with American Rivers. I sit in Asheville, North Carolina, but I lead our restoration efforts across the Southeast region. So a few updates. You'll hear a little bit more from me, and I have a cool video that I will share after lunch, but just a couple of high level updates for y'all that I want to go ahead and share. One is that to focus on some mussel research. So we are, to put it really simply, we're looking for states to adopt the Mussel Assemblage Health Index. I can pop, let me see, I'll pop up a one slide for y'all to see. And this is about the Mussel Assemblage Health Index. So American Rivers is part of a huge, over 100 organizations, over 13 states. It's international now. Shout out Alex in Canada. We're just trying to figure out why mussels are declining. I think everyone here is really acutely aware that that is what is foundational here. And we just need, honestly, basic biology to begin to look at that. So this tool, which again, I'll get into more lately, really looks at some basic how are mussels doing? Because a lot of times you're talking to your fellow state malacologists, how are your mussels doing? And it's not always super quantitative. So we're wanting to get a little closer to that. You can check out, I'll put in the chat a link to access this tool. And thanks to so many folks who are on the call who were a part of developing this and who are already adopting this. That's really great. Our aim is for state agencies to adopt this. And for a lot of states, it won't take very much additional work to do that. And it will give us the ability to track a lot of these declines. The other thing that I will let y'all know, a bit of a pivot. I spend the majority, even though my background is freshwater mussels, and I'm so glad to still be working in part on that. I spend the majority of my time working on dam removal projects and other aquatic connectivity issues. And unsurprisingly, we're often dealing with the presence of mussels at dam removals and other aquatic barriers. So there is a place for folks on this call to join these aquatic connectivity teams. I know Langston is a big part of the North Carolina aquatic connectivity team, and other people are as well. And so that QR code that is on that slide can take you to a site where you can join aquatic connectivity teams that are really all across the country. So you can sign up for any of those that are in your state. I'll put links in the chat to all of that. But I will get into a little bit more detail about MAHI, the Mussel Assemblage Health Index, later. And thanks so much. That's it for me.
Thank you very much.
All right. Hey, everyone. Can you hear me? Great. Awesome. Yeah. Hey, I'm Justin Lewandowski. I'm with the South Carolina Department of Environmental Services, based in Columbia, South Carolina. And also want to give a shout out to my coworkers, Zoe Riggs and David Ergel, who are signed in as well. And we do general aquatic macroinvertebrate sampling for water quality and stuff, but including with that is a good number of freshwater mussels. So we're always finding cool, a lot of our sites, we'll find, a variety of cool different mussels that we sample and, obviously release and note whatever we find in the field. But looking at specifics, we've gone on and helped with whatever we can, some either surveys or relocation projects, both to help out and also to increase our knowledge and learn more about, help us out with mussel identification and sampling and things like that. And so some recent ones, shout out to Erica from SCDNR. I know I've joined up with her and her group on a few surveys they've done. It's been really helpful learning more. And then also several it's perfect timing to actually segue into this, because talking about the a lot of the relocations and things that are done. So I know we've joined up with Tim Savage a few times recently for one was in the Congaree River and Canal right here in Columbia, a restoration project that dated back to the 2015 floods we had. We relocated a bunch of mussels there, and then in I know it was mentioned again, going back to the last few talks talking about dam removal projects, the Deep River in North Carolina. Zoe and I helped out with that one. One of the days collecting and relocating a lot of mussels there. So this is just great, the connectivity and everything like that. And it's great to get out and help out. And it's great for us to really help us learn more and more about mussels. And so I've really enjoyed doing all of that. And so, yeah, shout out to everyone. But I think that's a kind of a quick summary of what's going on here. So thanks.
Thank you so much, Justin.
Thanks.
All right, good to go. Mary, if you're ready.
Okay, cool. Hey, everybody. I think I know a subset of folks here. But yeah, by way of introduction, I'm with the Western Pennsylvania Conservancy and the Natural Heritage Program for the state. And so in Pennsylvania, we are a, delightful mix of resource agencies, and we partner, to manage freshwater mussels. So that includes the Fish and Boat Commission and the part of the Heritage Program in my organization. And then we have, I also have a colleague here in my organization who's from a different department. So he's going to speak about a project that has to do with the statewide mussel plan. But generally, our heritage program, keeps track of the tracked species for Pennsylvania and the data that goes into environmental review. So, we've traditionally done surveys for rare species, and now we also do some, conservation and management projects. So I'll mention that our state malacologist, Nevin Welty, has taken a different position at the Fish and Boat Commission. So his position will be open in the coming weeks. So keep an eye out for postings about that. We should be posting it on the Western Pennsylvania Conservancy's website. And let's see. So yeah, I'll give a few just sort of brief updates on some various projects. This year, of course, was the updates for the state wildlife action plans. So Nevin and myself and some other folks worked on updating the S ranks for the mussels in Pennsylvania, including, some species that are seem to be kind of coming into the state again in the Allegheny and Ohio rivers in Western Pennsylvania. And so we continue to do, I guess I'll also mention that we did do aquatic snail rankings as well for the Wildlife Action Plan. We haven't gotten yet to spherids or other mollusk groups, but hopefully one day. We kind of continue to do inventory projects for rare species, and so we do have a couple of those going on, mostly in Western Pennsylvania right now, so in the interior basins, but we do also do some projects in the Atlantic Slope. We are going, I am kind of participating in a couple partnership projects. One of them sort of parallels the Pennsylvania Mussel Conservation Plan, but this will be for the Upper Susquehanna Basin, and it's kind of in collaboration with the Upper Susquehanna Conservation Alliance. So it's the New York DOT, Fish and Wildlife, SUNY, some other, the New York, apologies, my brain, resource agencies. And so we did some inventory of mussels in the Upper Susquehanna as part of a data collection phase. And then we're going to be, summarizing data just on, some looking at threats to populations in the upper part of the basin and, mapping some areas that we see as needing restoration, areas that are conservation opportunities, and just trying to come up with some goals and next steps for that region for mussel conservation. And then I'll just wrap up with a project that some colleagues from Maryland and some other folks are working on with our organization in the West Branch Susquehanna. So that's a tributary to the main stem Susquehanna, kind of in central Pennsylvania, and that area's had a lot of mine drainage issues, and they've been slowly remediated over the years, but there's kind of a big stretch of the main stem river there that just hasn't had freshwater mussels for a long time, and it's upstream of some dams, so there's passage issues with host fish, and so even though the water quality is better, the mussels haven't been able to return on their own. So this project is funded through NIFWIF, and we're kind of in basically year 2 of the project, but the goal is to start stocking yellow lamp mussels and also American eels that are inoculated with Eastern Elliptios in these areas that have been the mussels have been excluded from since pollution. Anyway, so that project kind of got off the ground mainly last year. We did some phase 1 sort of steps to put mussels in silos and look at survival. And then we did put a few adult yellow lamp mussels out in some of the places that we plan to stock them to see how they did. We're analyzing that data, but it'll be kind of used to guide, where we stock yellow lamp mussels this year and eels again, but that's kind of the big push is to get a lot of eels and with elliptios and also yellow lamp mussels out. And so, yeah, we've been fortunate to, work with Maryland DNR and Matt Ashton and some other folks who are doing the propagation, and also our Pennsylvania Fish and Boat Commission and Susquehanna River Basin Commission, who's, catching the eels at the dams on the lower Susquehanna, and then we're using those to inoculate, and another colleague at Lock Haven University. So anyway, I'll just wrap up there, but yeah, let me know if you have any questions.
Thank you. Thank you. Let's see, who did we have next? Natalie Davis.
Hello. Let me share my screen real quick. Okay. Oh, oops. So hi everyone. My name is Natalie Davis. I am a master's student at NC State in the Marine, Earth, and Atmospheric Sciences Department. I am advised by Dr. Jay Levine, who is also on the call today, and I have Sarah Halliburton here with me, who is our technician for this project. So Sarah and I will be representing all the folks working on this project and sharing some of the progress that we have made so far. So we have a little mini presentation, but we'll keep it brief. So this project has a long name that I'll let you all read, but we have lovingly shortened it to BLISS, which stands for Bivalve-Based Living Sensor Systems. The goal of this project is to develop an early warning response system to contaminants in surface waters using freshwater mussels, multimodal sensors, and AI-based analytics for continuous monitoring of mussels' individual and group responses. Okay, so far our collaborators in the electrical engineering department have developed and built the sensors that include PPG and ECG sensors, and these collect heart rate, and this allows us to evaluate the physiological responses of the mussel. The sensor also includes an accelerometer that measures the gaping width of the mussel and a pressure sensor that can measure how far the mussel is burrowing into the substrate. And so this allows us to evaluate the behavioral responses of the mussel. And so these sensors are waterproofed, and then they're adhered to the mussel shell using dental cement. And they are currently wired, but the engineers are working on making them wireless. Another component of this project uses high-resolution cameras to record changes in behavior. Contaminant exposure studies will take place inside this bioreactor in the top right, with 4 surrounding cameras to record individual responses to chemical stimuli. Group behavior is being recorded in the living stream setup by an underwater camera and one overhead to help track movement. And then you can go to the next one. Okay, so we are going to be using the sensors to collect data to evaluate mussel responses to different algal food sources. So I have successfully cultured 3 different strains of eukaryotic phytoplankton, and we are currently in the process of feeding these to the mussels in 3 separate trials and collecting the data with the sensors. And then I will later culture 3 strains of cyanobacteria, and we will run a similar experiment with this. Following Natalie's feeding experiments, we will soon be moving to the contamination exposure experiments. These will combine the on-body sensors with the camera recordings. Initial exposures will measure elliptio responses to ammonia and manganese inside the bioreactor. They're also using the recorded videos for a citizen science initiative, and those will be uploaded to Zooniverse. And this is where volunteers will help train the AI algorithm in detecting movement as well as changes in gape and behavior. Ultimately, we hope to deploy these cameras and sensors at the inlet and outlet of public water infrastructure to be used as the early detection system. And that's all we have today. Thank you so much. Let us know if you have any questions.
Thank you very much. All righty. Next up, we've got Megan Kubala, please.
Okay. All right, hopefully that's up. Hi everyone, my name is Megan Kubala. With Matt Ashton and I, we make up the Freshwater Mussel Assessment at Maryland DNR. And basically, we do all things to support the conservation and restoration of freshwater mussels in Maryland, whether that's surveys, monitoring, propagation, translocation, and various activities like that. So today I'm just going to give a brief overview of one of the larger projects that we've been working on, similar to the silo study that Mary was talking about with the folks in Pennsylvania. We are looking at restoring freshwater mussel populations to the upper Potomac River. It's an area that even though the water quality has improved, the mussels aren't recolonizing on their own. And we just want to see what the potential is to restore that functional population there. So this is a long-term study. We started in 2024, where we floated about 70 miles of river, and we mapped all suitable patches of habitat. And then last year in 2025, we did our silo study where we looked at growth and survival rates of eastern elliptios. And then at the end of that, we started some small scale stocking plots to do long-term monitoring of those mussels. And we are currently now in the phase where we are drafting a restoration plan based on our findings. And so just briefly, we conducted last year 32 surveys using the Brook Floater Rapid Assessment Protocol just to see what mussels were there in that area. And then we also did a silo study where we put out 30 silos at 10 different sites, and then we monitored their growth and survival every 30 days over 90 days. And then we also took water quality samples along with that to see if there's anything limiting their growth. And so, not surprisingly, we saw that our mussels grew fastest during that warmer months. So you can see from July to August, they really took off. And then as the water started cooling down, their growth started slowing down also. But then more interestingly, we did see that the mussels further downstream in our study area grew significantly greater than the mussels that were further upstream. And so looking at our water quality, we did a PCA of 19 different water quality parameters, and we found that nutrient availability explained the most variation across our sites. And we also saw that our PC1 scores were positively correlated with mussel growth. So we could see that there's definitely something going on with the nutrients and what those mussels are eating. And so just taking this back big picture, we had about 97% survival across all of our sites. And so that's really encouraging. So we didn't really find anything that's affecting their survival, but seeing that there is a gradient in growth across the range of that area, it kind of highlights the need for more data related to primary productivity and also maybe sediment chemistry to see, what's in the ceston that those mussels are ingesting, and then also, to see if there's anything in the sediment that could be inhibiting their growth as well. But yeah, that's pretty much the updates that I have. We work a lot with other local, state, and federal partners. A lot of folks are probably on this call. But yeah, that's all I have for today.
Thank you so much. All right, up next we have Joe Nichols, please.
All right, everybody. My name is Joe Nichols. Can you hear me? Good.
Yeah, you're good.
Sweet. So yeah, my name is Joe Nichols. I'm a biologist with Virginia Tech at the Virginia Fisheries and Aquatic Wildlife Center at Harrison Lake National Fish Hatchery in Charles City, Virginia. And so this operation is kind of a joint effort between U.S. Fish and Wildlife Service, Virginia DWR, and Virginia Tech. And so we raise about a dozen different species of Atlantic slope mussels, and we also do some river herring restoration. And so in total, since around 2008, we've released around 420,000 mussels and delivered around 300,000 mussels to various other state agencies, universities, and NGOs. So in 2025, we released around 63,000 mussels, which is it's either our second highest or highest release amount. And those are, that's just for what we've kind of put out in Virginia. And then we've delivered this year around 30,000 mussels to other partners. So through partnerships with River Association, Maryland DNR, North Carolina Wildlife Resources Commission, partnership for the Delaware Estuary, and Anacostia Watershed Society, and obviously U.S. Fish and Wildlife Service and Virginia DWR. In 2025, we've been able to kind of distribute mussels across Virginia, Maryland, North Carolina, Washington, D.C., and around the Delaware Bay. So I think that's pretty cool, just the amount that we've kind of been able to spread around. We've kind of also been able to be involved with producing mussels for mussels in the classroom. So and giving mussels away to some high school classrooms. So just trying to get those mussels in the public eye a little bit, especially with the younger people. That's been really kind of a neat experience. And we're also doing some cage studies, so also working with some volunteers on that. I think that's pretty neat. So propagation wise, some things I want to focus on for things that we've been able to produce this year. These are just some highlights. We kind of did a lot of species, but Elasmodon varicosa, brook floater. I think Brian's going to talk about that a little more later, but we actually had a really good year with production. And there were a lot of just kind of like little things that we kind of worked on, like stocking density, moving them out into wild water, like finding the right timings for things. And I think we kind of nailed it in terms of having high survival and good growth for the first year. So I'm pretty proud of that. And then we also did a lot of yellow lamp mussel, and that went really well. So we're hoping to do more of that in the future. And then Elliptio campanata, we've kind of had a few hit or miss years, and then this year we had a lot of drop offs using brook trout. So seeing that kind of be successful, that was another thing where we just kind of like figured out all the right parameters. So we're hoping to kind of replicate that again in the next year. So yeah, pretty good stuff. I think Brian is probably going to be able to expand a little more on some of that, but yeah, that's all I have for now.
Thank you, Joe. Yeah, I've got Brian coming up in the next group if that works out for everybody. If we can continue though on this, let's go ahead with Andrew Rapp and Allison Orcutt.
Can everyone see the screen, the presentation right now?
Yep, we've got your presenter view.
Okay, perfect.
See it. Andrew, I'll go ahead and start, and I'll introduce myself, and then I'll let you take it from there. So hello, everyone. I'm Ellison Orcutt. Myself and Andrew Rapp are from the Virginia Natural Heritage Program. We're nestled into the Department of Conservation and Recreation in Virginia. So we aren't state wildlife agency. That's DWR, but of course we have a lot of interest in rare species. And so I just put up here a list of the staff that we have that have contributed to or currently contribute on mussel work in the state. Ann Chazal used to be a field zoologist for our program. Now she's our chief biologist, so she's an office person mostly now, generating funding for us, but she used to do mussel surveys for us, and she still does a little bit. Steve Robley is in a largely office-based position now as he nears retirement, but he's done 30 years of mussel surveys in Virginia. And myself, I'm the lead of two field zoologists that are relatively new to our program. Both started in 2024 and are really getting their feet wet, literally, with mussels over those last 2 years. Those are Andrew Rapp, who will speak with you about one of his projects, and Derek Hennon, who has contributed a little bit to our mussel work. So, Andrew, if you switch to the second slide there. So just a brief overview of our general efforts in the state before Andrew highlights on one specific project. Our Natural Heritage Program has been in existence for 40 years, and we have done mussels most of that 40 years. However, we are an agency or a division that's interested in all rare animal species in Virginia, and so we try to be a jack of all trades. And so mussels are not a primary focus of ours. They are, I would say, on any given year, anywhere from a quarter to a tenth of the field effort that we exert on rare species in Virginia are on mussels. But we have tried to make it a point in the Atlantic slope to maintain some active field survey for a T&E species most years, despite the mussel diversity, of course, in the Tennessee drainage, which Virginia gets a nice sliver of. We don't do any work out there. Manpower, partly the fact that it's further west than Detroit doesn't help when you live in the eastern part of Virginia. And also, it's just a diverse fauna that we haven't had a chance to really learn. So we've just focused on Atlantic slope species. We're fortunate that Brian Watson and DWR have been great partners to us in mussel work. Of course, Brian will speak a bunch about that later, about his own work later. But we depend on funding. Both Andrew and Derek are not state-funded, taxpayer-funded zoologists. We generate grants to fund them, and oftentimes Brian and the folks at Fish and Wildlife have given us Section 6 funding to survey for species you can see listed there: James spiny mussel, Atlantic pigtoe, yellow lance, green floater. Those have been our main focal species for projects over the last 30 years. And a couple other rare species that are in the Atlantic slope that we're interested in, but that we haven't spent much time on, or just maybe not that relevant as T&E. But yeah, I'm excited to learn from everybody's work and excited for Andrew to share one of our current projects.
Good morning, everybody. It's nice to be here and listen to all the awesome projects that are going along in the Atlantic area. My name is Andrew Rapp. I am one of the field zoologists for the Virginia Natural Heritage Program. And this past year, we conducted a first of 2 years of work in the Cowpasture River, which I'll explain where the Cowpasture River is shortly. But the main focus of this project is hoping to connect with James spiny mussels. The Cowpasture River is located in the central west mountains of Virginia. It flows in Highland, Bath, Allegheny, and a tiny bit into Botetourt County in Virginia. This river has a history of having James spiny mussels, but overall very minimal amount of surveys in the river. So it's a main target of ours to get a good number of surveys out in the Cowpasture River and then hopefully connect with James spiny mussels. So the Cowpasture River flows and connects with the Jackson River, and then around Clifton Forge area begins the James River, and it is within the James River watershed. Here's some of our results from the field season. We were able to connect with a total of 20 James spiny mussels. Funny enough, 18 of those individuals were those released and they had pit tags. So we got to see some of the efforts of Virginia Department of Wildlife Resources in action in the field. But we were able to also find 2 unmarked individuals at a new location. We also found a good number of Venustaconcha constricta, notched rainbow, Strophitis undulatus, the creepers, only a few Eastern elliptio, which was quite a shocking surprise, having done work last year in the Meharon River where we found thousands of them. So it was very funny seeing only a few in this system. And then 40 Elliptio fisheriana complex. Here's some pictures of some of the mussels we found this year. The bottom left being some Strophitis undulatus. Bottom center is a newly found James spiny mussel. And on the right, we have a nice comparison of Venustaconcha constricta on top and a James spiny mussel below. And this is the year 1 of year 2, so we're looking forward to getting out again next year. And with a little better of understanding of best areas to target in the Cowpasture River, we're hoping to have more success next year. And this is just our contact information. Though Loretta beat us to the punch there, we've already got our information on that great spreadsheet. So if you're looking to get in touch with Allison or I about our work, feel free to reach out. And
I have to finish with a funny story here, Andrew, about this picture. I'm always looking for a chance to crack a joke, so I apologize here, but that's me, the rest of me besides my head that you see now. But we had a project for Atlantic pigtoe a couple years ago, and the Meherrin and Nottoway River systems. And if anybody that's done a bunch of mussel surveys, I'm sure can appreciate the mussel treasures that they find that are not mussels, but things that now litter their dashboard or their desk or their bookshelves. And on a day where we had bet that the next person to find a Atlantic pigtoe, because we were having trouble finding them, would win a prize, I found an Atlantic pigtoe, and I got back to the truck before Andrew. And on his way back, he had found a discarded over the bridge trophy for the 16th place in the pistol shooting championships of the local hunt club, which is now my Atlantic pigtoe trophy from that field survey, and sits on my bookshelf. Sorry to share. 16th place trophy, though. I thought it meant something.
That is amazing, and I'm very glad you shared. That's probably what we should share is like, what's the oddest thing you found? 16th place is amazing. Thank you. Alrighty, next up we have Annie Stupik, please.
Hi everyone, my name is Annie Stupik. I'm with the New York State Department of Environmental Conservation. So yeah, New York, it's kind of this odd position, right, where we have the Ohio drainage species and then we have Atlantic slope species. We have about 44 species in the state, and we're not anywhere near like you guys in Virginia, right, and the Carolinas, but we're also have more than the New England states, right? So unfortunately, the agency hasn't had either in a natural heritage program or in the state agency, which is where I work, we haven't had that much effort or sort of focus internal capacity for a mussel program statewide. But luckily that has changed as they hired me, and then my partner Kelly Crandall, who's on this call. We have at least a good chunk of our time devoted to mussels. So yeah, we were able to devote a decent amount of effort to our species status assessments for our state wildlife action plan. And we're sort of at the beginning of this exciting and challenging phase of trying to work with folks, both in university partners, New York State Museum, other folks in our own agency who are doing a lot of great restoration work, to sort of try and create a mussel conservation program across the state. So yeah, I think, let's see, I can let my partners speak up on their own work if they want to chime in. But right now, what I'm working on is some of our state wildlife action or state wildlife grant, projects. And so one of those is planning some baseline surveys in the eastern part of the state, which is really undersurveyed. So we'll be targeting lots of species, including dwarf wedge, green and brook floater, alewife floater, tidewater mucket. We're also pretty concerned about eastern pond mussel and margaritifera from the state perspective. So we'll be targeting a bunch of species for those surveys, which will hopefully get done in the coming years. And various partners in the state we have working on different mussel projects, including eDNA in the western part of the state, targeting salamander mussel and snuffbot, as well as survey and restoration efforts in some of our legacy contamination areas. So yeah, I guess just for a quick plug, what we're hoping to develop is a framework for restoration efforts regarding release of propagated individuals into the wild. I do not have a background in aquatic ecology, so I think we've got a decent idea on like the ideal situation for the genetic requirements as to where you're sourcing broodstock. But if anyone has any SOP on disease screening for releasing propagated individuals into the wilds, that would be really helpful. We're sort of looking to develop an SOP on what we'd like to test the water where the mussels are coming from, invasive screening, things like that, before allowing folks to release into the wild. So I'm going to put my my email into the chat if anybody would be willing to send their state protocols or whatever protocols your lab uses. That would be great. So yeah, I think that's all I've got. Thanks for organizing this. Great to be here.
Yeah, thanks for joining. That's what I'm trying to say. Hopefully there's some folks here that can help you out with that. So that's great. Speaking of volunteering and stepping up, we are just about at the end of my list. So here after Max is the end of the list. So if I missed you somehow, I'm very sorry. Please let me know and I will add you to the list. We'll keep going. So call for last minute volunteers. And while y'all message me with that, let's do Max. Max Stridell, please.
All right. Hi, everyone. I'm going to share some slides. So my name is Max Streedel. I am with the Florida Fish and Wildlife Conservation Commission down in Gainesville, Florida. And my update today is about a new project that we're getting started on, which is the propagation of Medionitis walkeri, the Swanee moccasin shell. So for those of you unfamiliar, Swanee moccasin shell is endemic to the Swanee River Basin, and it's federally threatened under the Endangered Species Act. It's a small mussel, only getting to a maximum size of around 53 millimeters. And it has some known hosts are sooty-banded darter and brown darter. Mainly sooty-banded darter is the more effective host for this species. And sooty-banded darter has recently been split from the black-banded darter. So Medionitis walkeri has been extirpated from a large portion of its historic range. So it's been extirpated from the Withlacoochee River, that this top circle, and also from the upper Santa Fe River. So our goal is to reintroduce Medionitis walkeri into these places where it's been extirpated from, and we're going to be doing that with in a stream-side propagation trailer that will pump water directly from the Santa Fe into the trailer. And so we are currently just waiting on getting that trailer to our site, and that should be in place by the end of the month. And then we can really get going on getting the plumbing and getting that trailer set up so that we can get some juveniles going. And we're currently acquiring host fish. So we have a darter tank up and running in Gainesville where we have 35 sooty-banded darters and a couple brown darters as well. And then our colleagues in the Florida Panhandle also are accumulating some darters. So our goal is to have a March inoculation this year with a small number of Medionitis walkeri broodstock, and then also some Lampsilla straminia southern fat mucket broodstock as kind of a common hardy test species. But yeah, feel free to email me if you're interested in this project or want to collaborate. And then I'm going to also pass the laptop over to my colleague who's also doing some Medionitis walkeri work.
Hello, I'm Allison. I also work for the Florida Fish and Wildlife Conservation Commission here in Gainesville. As Max talked about, the Swanee moccasin shell is a federally threatened species. So my current project is a Swanee moccasin shell species distribution model. I'm looking primarily in the Swanee River mainstem. The objectives of the study are to identify important variables that drive distributions of the Swanee moccasin shell, and determining these variables could be helpful when we're locating suitable reintroduction sites and also identifying suitable habitat within the current range for population augmentation. For methods, we're using the R package SSDM, which has options for creating stack distribution models as well as ensemble species distribution models. I've been working primarily with creating ensemble models as they combine multiple models to improve overall accuracy. I'm looking at a few abiotic and biotic factors as potential predictor variables. For abiotic, we're using substrate maps derived from side scan sonar surveys. We have water quality data, and we're also using cumulative springs discharge. The Swanee River is fed by many springs, and some past literature has found a correlation between the springs discharge and the walkeri distribution. For biotic factors, we're looking at host fish biomass. As Max mentioned, they primarily use the sooty-banded darters. So we're using long-term monitoring electroshocking surveys to make a darter biomass layer to use in the model. This is my primary project at the moment, but I've also worked on calculating the Mahi scores for rivers here in Florida. So if you have any questions or want to talk about species distribution models, my email's down here.
All right, thanks.
Thank you, thank you. All right, Lindsay Zarecki, please.
Hello, I'm Lindsay Zarecki. I'm the Vice President of Conservation and Research at the Greensboro Science Center here in North Carolina. And I work with my team to do a handful of freshwater things, including we have a long-term study site where we are looking at the impacts of mitigation and how that affects different taxa. And that does include our freshwater mussel species as well as the fish that we have in our streams. So we've been monitoring that and it's ongoing. And then the other thing that I am going to be talking about a little bit more later is that we are part of the Association of Zoos and Aquariums, which is about 250 accredited institutions in our country. And they have a flagship program called SAFE, which is Saving Animals from Extinction. And over the past year, we have worked extensively to produce a freshwater mussel program plan that aligns with the Freshwater Mollusk Conservation Society. So that has been quite an effort with a lot of people, and I will be talking about that more later. But one of our goals is to get to know everyone else who's doing conservation work with freshwater mussels. So it's wonderful to be here today and to hear all these projects and know that there's a world of opportunity out there for collaboration. We also have a freshwater mussel propagation lab at our zoo. However, my technician is on this call and she maintains that space. So I'm going to let her go ahead and introduce herself and talk a bit more about that.
Hi, everybody. My name's Hannah Damewood, and I'm the lead conservation and research tech at the Greensboro Science Center. I'm going to share a couple slides that I have prepared. Let's see. Okay, can everybody see that? Okay. So this is just a little intro slide I made about me. This is me holding some Velosa dolumbus that we caught out on our large mitigation project that we do surveys on every year. So for our propagation lab here at the Science Center, we have been focusing on Velosa dolumbus, but we're also starting to work with Velosa constricta. This December, we were actually able to release about 200 individuals over in Liberty, North Carolina. And those individuals were propagated in the spring of 2024. And I'll share pictures from that in just a second. And this past summer, we collected fantail darters for our Velosa constricta propagation with the WRC here in North Carolina. And then we also collected Velosa constricta with the WRC and Mike from App State. So we're super thankful for all of that help and the partnership that we have there. And we're hopeful to have a really good, successful propagation with that species this spring. And then let's see here. And then this is just a handful of photos. We had one of our photographers from the Science Center come out with us during our release of our propagated mussels. And these are some of the folks that were with us. We were very thankful to have Langston and Brina and Jacob out there with us releasing these mussels. And we were so excited that we were able to share this with our community and do a social media post with it about from the Science Center, and it got a lot of really good traction. So very excited about all of that. Let me see if I can stop.
All right, thank you very much, Lindsay and Hannah. Brian Watson.
Good morning. My name is Brian Watson. I'm the state malacologist for Virginia Department of Wildlife Resources. Been with the agency for going on 24 years. I cover all things freshwater mussels, for the state, also do snail work, sphaeroid work, get into crayfish work as well, a little bit of terrestrial snails as well, but primarily freshwater mussels. I'm going to talk about some brook floater work that we've done, but just kind of give an update of the mussel team here in Virginia. Probably for about 20 years, it was basically myself and a part-time tech who were hired through DWR. And then we kind of worked through a number of partners. As you saw with Ellison and Andrew, we'd contract a good bit of work to Heritage and other partners like TNC and Virginia Tech. But here over the past few years, we've hired on some new staff to kind of flesh out the mussel team here. We hired a full-time assistant state malacologist about 4 years ago, and that position is currently manned by Zach Taylor. He's been in the position for about a year. And then we hired a couple freshwater mussel biologists, Kim Morgan and Kayla Howard, and they were primarily brought on to put together a statewide freshwater mussel conservation plan. They were brought on about 3 years ago this spring and summer. So they've been working on the plan. I know Kim will give an update on that later this afternoon. And then once the plan was developed, they would transition into help implementing conservation and recovery activities. And I've done most of my work in the Atlantic Slope because we've had a lot of partners in our freshwater mussel hatchery down in Southwest Virginia doing work down in the Upper Tennessee for 20, 30 plus years down there. And there's been a real kind of shortfall on the Atlantic Slope side. So that's where I do most of my work, but I still do some down in Southwest Virginia. And then we've got the mussel propagation facilities. You saw Joe give a talk earlier for Harrison Lake National Fish Hatchery, the Virginia Fisheries and Aquatic Wildlife Center. That's a cooperative facility between DWR and Fish and Wildlife Service that propagates mussels on the Atlantic Slope. And then we have the facility down in Southwest Virginia as well. I'm going to share a slide. I don't have many more than this one, but those of you who were on the brook floater working group call about a month ago, this will be familiar to you. Let me know if you guys are seeing that. I'm not going to put it in presenter mode or anything. I'll just give you an update as far as what we've been doing with brook floater here in Virginia for about the past 7 or 8 years as part of various projects. For us, brook floater is a state endangered mussel in Virginia, believed to be extirpated. Last time it was seen live was 1998. Its range in Virginia is primarily up in the Potomac and Shenandoah watersheds. In Northern Virginia, we have a single record in the main stem James River. And if anyone's familiar with the Potomac and the Shenandoah, heavy impacts from development up in Northern Virginia and then agriculture throughout the Shenandoah Valley. And so as part of that brook floater working group, we had a comp swig grant, 2 of those that Allison had mentioned earlier during her talk, and that kind of funded a lot of this initial work, trying to flesh out the propagation, which host fish work, kind of techniques, best techniques for growing out the mussels. And this slide just shows the propagation number since 2018. That middle column shows how, in the parentheses, is how many gravid females we use, and then the host fish we were using in the right column. So 2018 was kind of the testing for us, and we did a number of different fish, brook trout, margin mad tom, long-nosed days, sculpin, variety of Luxilla species, and fantail darter, and golden shiner, and also hatchery brook trout. You kind of see those numbers there that we got from some of those fish. That mix of that first row there, most of those 3,217 juveniles came from brook trout. I think it was a dozen came from those other species. And so over the years, we've really focused on brook trout for producing our numbers. We can get those easily at one of our DWR hatcheries. Golden shiner has worked off and on for us. That first year it worked really well. Those were golden shiners, larger ones we got out of one of our ponds, but ever since then our numbers have been really unreliable for golden shiner. So we focus on brook trout. You can see we produced over 150,000 since 2018. I know when I gave this talk at the brook floater working group, meeting, Michael Perkins had asked about what was their survival of those animals, how many made it out the door. I don't have a slide on this. I kind of threw numbers together. But the goal with producing these was actually to start releasing the animals back to the wild. To reintroduce brook floater back to Virginia. And so we've done that as part of the Comp SWIG project. The second grant we got was more focused on that. And then we also have a natural resource damage and assessment and restoration settlement through DuPont, where we're doing mussel restoration work in the South River and South Fork Shenandoah River watershed. And brook floater is one of those species. So between those two projects, we've released, and these are taggable brook floaters, so you're talking 20 millimeters plus in size, just shy of 7,700 animals. That's about 3,500 for the Comp SWIG project and about 3,250 for the DuPont NERTA project that have gone out. For the Comp SWIG, that's been about 4 streams at 8 different sites, and the DuPont project has been 2 streams, the South River and the South Fork Shenandoah, at 8 different sites. And so that's about a 7% success rate from what we've produced to what's gone out the door. As Joe mentioned, some years we've had better success than others. This year we had really good luck with kind of honing in on some different things that we're doing at the hatchery that really upped our survival. So I'd anticipate those numbers going out next year and the year after from a 2025 stock will probably be more in the line of 10% plus as far as the survival of those animals. And then we've also given the partners over the years, we've worked with Matt Ashton at Maryland DNR to provide them with about 500 brook floater for some restoration work. And then given some back to West Virginia as well, because that's where we get our brood stock from, is the Kakapo River, which is a trib to the Potomac River. And that is all I had for the brook floater stuff we're doing in Virginia.
Thank you very much, Brian. Up next, we have Trent Mellum.
Hello, everyone. My name is Trent Milam. I work for the Watershed Conservation Program at the Western Pennsylvania Conservancy. So you heard Mary talk a little bit earlier about some of the stuff we've been working on and that she's been working on with the Natural Heritage Program. The project I really wanted to highlight was a huge project we completed in 2025. WPC, along with the Pennsylvania Fish and Boat Commission, completed the Pennsylvania Freshwater Mussel Conservation Plan. This was a big endeavor that was tasked to us by the National Fish and Wildlife Foundation. They wanted to see a mussel plan developed for the state of Pennsylvania. Our first step throughout this project was working with the Fish and Boat Commission and the Natural Heritage Program to organize all of the known freshwater mussel data in Pennsylvania into a single database, which was a huge undertaking. Thankfully, Mary had already done a lot of that work leading up to around 2008 in a previous project. So we continued from there, and together we kind of organized a database that we now have for historical all the way up to 2024 as of now, freshwater mussel records in Pennsylvania. And then our goal for the actual mussel plan was to make it a comprehensive document, as comprehensive as possible. So we didn't want to focus on any specific species or group of species or any particular location necessarily. And so we did this by assessing historical and current mussel trends throughout Pennsylvania and each of the 6 major watersheds and also all of the Huck 8 watersheds, which was kind of our unit of organization for the plan. So there's a chapter in the plan for each major basin and then a section for each Huck 8 watershed. And each of these sections, we look at the mussel data, we discuss threats and impacts to the watershed's mussel populations, and then we put forth recommendations for conservation moving forward. This document also has a pretty extensive background section and some other chapters as well. And we also, as a part of this project, developed a watershed prioritization for Pennsylvania. So we tried to identify the highest priority mussel watersheds and streams for protection, restoration, surveying and monitoring, and then propagation, augmentation, and reintroduction efforts as well. So some of that's the places we're already doing that work in Pennsylvania, like the West Branch, and then some of it is places that we would like to see more of that augmentation work happening in the future. And the criteria we use to develop all of these priorities were data-driven and jointly decided upon by everyone involved in the project. So, I mean, overall, we're really excited about this mussel plan as a tool that we can use moving forward, that conservation practitioners can use to seek funding in Pennsylvania with mussels in mind, or to use as a guide for all things mussel conservation moving forward in Pennsylvania. We also were attempting to kind of sync this up with the timeline of the State Wildlife Action Plan. So we are hoping to update this mussel plan every 10 years to kind of coincide with that. The plan itself is finished and was completed a couple months ago, and we're just in the process now of getting everything out to the public and putting everything online, and hopefully, WPC that's going to happen in the next couple of weeks. So that everything should be available to the public soon, and there's also going to be an interactive online GIS experience that goes along with the mussel conservation plan, where people can take a closer look at some of the data that we have used and are using. We had so many project partners involved in this, as it was obviously a large endeavor. So many thanks to obviously the Fish and Boat Commission, the Natural Heritage Program, Maryland Department of Natural Resources, Pennsylvania Department of Environmental Protection, the Susquehanna River Basin Commission, American Rivers, and many more as well. And that's kind of the main project that we had been working on last year, and that's all going to be available soon for anyone who's interested to explore it and use the conservation planning, not only for Pennsylvania, but hopefully as a tool for other states that can get some inspiration from it as well. And outside of that mussel conservation plan, we've been kind of like Mary said, just filling in survey gaps throughout Pennsylvania. It's one of the things we've really been focusing on, is finding places that are undersurveyed and going out and trying to document mussel presence and absence, and doing some more targeted surveys in the Ohio River Basin in Western Pennsylvania, specifically looking for round hickory nuts in the Shenango River, and some other projects that we're doing. So that's all of the update that I have.
Thank you so much. All right, last up for this group, I have Paul Stanley, and then we'll take a quick 5-minute break again, do a little stretches real quick, and then we'll finish up the last of our introductions. Thank you. Off to you, Paul.
Hello, everybody. My name is Paul Stanley. I'm currently a master's student in conservation biology at Central Michigan University. My advisor is Dave Zanata, if you're familiar. And my graduate thesis is on sort of the range-wide and genome-wide SMP-driven analysis of the phylogeography and genetic variation of the yellow lamp mussel. So the two sort of objectives I have with this project are to, one, construct a phylogeny of Lampsillus caryosa, the yellow lamp mussel, and other closely related Lampsillus species. And this is just to sort of confirm phylogenetic relationships between Lampsillus caryosa and the other closely related species. And our second goal is to determine genetic diversity within and among river drainages across its distribution, and to sort of assess geographic patterns. So we're really just looking for like populations with distinct or unique genetic variations, or to determine if the species is in fact panmictic, but I doubt that. And hopefully with this project, we're going to be able to start defining management units genetically. We're also trying to determine which watersheds are candidates as sources for relocations and / or hatchery propagation. And the last part's mostly for me, but I'm interested in determining the effects of glaciation on the yellow lamp mussel population, and seeing if I can determine the routes of recolonization from refugia to the sort of current populations we see now in the northern Atlantic slope. So we got our samples from all over the Atlantic slope, so I'd like to thank anyone here that we've partnered with to sort of send us samples for processing. In total, we have 152 yellow lamp mussel samples, and that's a part of 380 total samples, including all the other closely related species. Um, so, oh, and a part of that is that we also cover about 10 different watersheds. Now, I don't have too many updates on this right now. This past summer, I extracted the DNA from all the samples, got that amplified and into libraries, and we submitted the libraries actually not last Friday, but the library before to get sequenced. And we actually just got confirmation like about an hour ago today that our library is going to be sequenced, which is very exciting. Through the QC, we saw no real problems with the library, so everything's going very smoothly. And I should have the sequenced raw data back on Monday. So after then, hopefully I'll be able to get some analyses done, get some preliminary results sooner rather than later. I think that should be about it.
Fantastic. Thank you so much. Let's take a quick 5-minute break again, do some jumping jacks or something, get blood flowing. And here's another discussion question based off of the 16th place trophy. So what's the most interesting thing you've found in the field? And I'll see y'all back here at about, just 20 after, just a little bit before. Thank y'all. Alrighty, welcome back, everybody. This was a fantastic chat. I think a lot of us have found some of the same things, and that's great. I'm a little jealous about some of it, so cool. Y'all can finish adding to that as you feel that you want to, if you missed it over your short break. And when Kelly is ready, we'll jump back in. We'll give her one more minute or so.
Loretta,
Oh, perfect.
Kelly just messaged. No, sorry, I'm not Kelly. I'm Annie, her friend or colleague. She just messaged you. She said she lost power for randomly. So
oh, no.
she'll be back on in a bit.
Okay, if you let her know that, we'll just we'll add her to the end of this. We'll just move her to the end of this little section, and then we'll carry on. Thank you so much. I don't think the message came
only
through.
for 30 seconds, but yeah, so she should pick out in this second.
Oh, 30 seconds does a lot of damage on your computer. Not damage, but like now you have to open everything, you know. Puts you off your game. Okay, well then we'll just pop Kelly down to the end of the section, and we'll go ahead to do Susan and Anna if you guys are ready.
Sure. Hi, I'm Susan Gita. No problem. I'm Susan Gita, and this is Anna Rainey. I'm just going to introduce myself really quick before Anna provides an update. I'm the project leader for the Freshwater Invertebrate Conservation Group within Florida Fish and Wildlife Conservation Commission. Lauren Patterson is actually the mussel program coordinator, but unfortunately could not make it today. But the rest of our mussel biologists did, they were able to attend to give some updates on our ongoing projects. That includes Allison Achia and Max Streetel and Anna Rainey, of course. So our projects involve everything from long-term monitoring to natural infestation research to propagation for the recovery of listed species, as Max and Allison covered earlier today. We only have several basins in Florida that drain to the Atlantic, but we do survey them as part of our routine monitoring. And Anna is going to provide some more detail on that. And I'll let her take it away. See if I can share my screen. Which one doesn't want to do? Don't know what sharing that one. Okay.
We've got your presentation now, or we had it for a sec there. Yeah, we got presentation view on it.
Oh, do the other one. Sorry.
No, you're good. There we go.
There you go.
That was good.
Thanks.
Okay, so yeah, I'm Anna Rainey. I work for the Florida Fish and Wildlife Conservation Commission, and I'll be introducing our state's freshwater mussel monitoring program. So the Freshwater Mussel Conservation Program is part of Florida's Freshwater Invertebrate Conservation Group. Susie, Lauren, and myself are stationed in Milton, and our survey work primarily covers the western panhandle rivers. And then Jacob, Allison, and Max, stationed over in Gainesville, primarily work across the eastern panhandle and the peninsula. So the goal of the program is to establish baseline mussel distributions with exploratory surveys and establish a network of long-term monitoring sites across the state in each of the major drainage basins. So in each basin, we first do timed qualitative surveys to get a species inventory and water quality throughout the river, and we have temperature loggers throughout the rivers. Then we choose a subset of those sites to revisit for long-term monitoring. So for long-term monitoring, we use transect surveys with quadra excavation to quantitatively sample mussels and characterize habitat. And then we'll also conduct a qualitative species inventory and collect water quality data for the site. So since the program began in 2013, the program has surveyed in 19 major drainage basins and established long-term monitoring sites in 9 basins. The program has conducted over 1,300 surveys, including 131 long-term monitoring surveys,
On the Atlantic coast, we have monitoring sites in the St. Mary's River and a tributary of the St. Johns River. And our goal is to establish 5 long-term monitoring sites in each basin and visit these sites at least every 5 years. The program also conducts research into mussel ecology, particularly life history. So a article was published a few years back to help provide standardized methods for characterizing the reproductive status of female mussels, as well as the different stages of larval development. So we'll also assess gravidity at our survey sites, and that data has been compiled into a publicly viewable repository called the Freshwater Mussel Gravidity Almanac. And I'll drop that link in the chat as well. This allows us to display and receive data from others working in the area on the reproductive timing of freshwater mussels in Florida. And then we can use this data for projects such as host trials and propagation. Again, I'm Anna Rainey, and our program coordinator is Lauren Patterson. And I've got our website and contact information here. That's all I have. Thank you.
Thank you so much. All right, next up we have Koa Yamashita.
Oh, there. Can you hear me?
Yep, we got you now. Thank you.
Okay, sorry about that. So my name is Koji Yamashita, and I work for the Pennsylvania Fish and Boat Commission. And I oversee a new division within the agency called the Division of Aquatic Species Restoration. And really today I just wanted to talk about that briefly and just let everybody know that we're active now in what we're doing. So the division has been tasked with the propagation and restoration of non-game species, concentrating on state and federally listed species. So our work is primarily guided by our state wildlife action plan, and we do a lot of mussel work, so that's also going to be guided by the PA mussel conservation plan that Trent presented on a little while ago. And while the division was formed in 2025, so last year, it is, like I said before, relatively new, but it does include our Union City Aquatic Conservation Center. This is primarily a mussel facility that's located in the northwest part of the state and concentrates on restoration efforts in the Ohio River Basin. The facility has been propagating mussels since 2019. I'm relatively new to the world of mussel propagation and restoration. However, I do recognize a lot of names, in this group, and it's probably through your interactions with the Union City Aquatic Conservation Center and their staff, Scott Ray and Josh Arnold, who are our permanent biologists there. More specific to this group, I just wanted to let everyone know that we are in the process of designing a new facility. It'll be our Huntsdale Aquatic Conservation Center, which will be located northeast of Gettysburg in the Susquehanna Basin. And this facility is going to be concentrating primarily on Atlantic slope species in terms of mussels and restoration efforts in the Delaware and the Susquehanna Basin. So that's really all that I wanted to say. Just introduce myself, let everybody know that the division is out there. We're going to be doing mussel work along with other species of concern. And because we're still in the development stages at the Huntsdale facility, we're really just looking to start developing these collaborative networks, joining various work groups, and really getting a better understanding of what everyone is doing, especially with mussel propagation. And hopefully in coming years we'll be able to have some presentations and be able to show you guys what we're doing at that facility. Also, we're still in the process of hiring staff for the division. We'll have in the next 2 years 3 more full-time biologist positions coming on, and since the work will have a large mussel component, I just wanted to put it out there for everybody on the group, and for any students or anyone be looking for work in the field to keep an eye out for positions. And with the Pennsylvania Fish and Boat Commission, we'll be hiring people, and having the mussel experience will be great. So that's it for me. It's real quick. And if anybody has any questions about our program or about mussel propagation in Pennsylvania, feel free to reach out to me. As I mentioned before, I think a lot of you have had interactions with Scott Ray and Josh at Arnold at the Union City Aquatic Conservation Center. To reach out to them too as well. And thanks for the time. And so far, this has been great. It's really great just to see what everybody's doing throughout the region.
Thank you so much. I'm so glad you could join today. And apologies again for butchering most everybody's name on here. I'm trying my best. Hopefully I won't get it wrong next year. Up next, Caitlin Carey.
Hey, I'm Caitlin Carey. I'm a researcher at Virginia Tech in the Fish and Wildlife Department. For the most part, my projects are field-based assessments of species statuses and distributions across Virginia, Tennessee, and a little bit of Maryland. Across even, the upper Tennessee drainage, New River, and Atlantic slope drainages. Assessments are including also long-term monitoring sites for population dynamics of wild populations and post-release monitoring of reintroduced sites. A couple of projects we've completed more recently in the Atlantic slope. For an update, we did a reassessment of the status of yellow lance across the Rappahannock Basin, over 60 sites, as well as a marker capture study. Another one is the Brook Floater project with DWR that Brian Watson actually just gave an update about. We were doing the rapid assessment surveys, is also monitoring some of their release sites of their captively bred mussels using a biomark floating antenna. And a couple updates on ongoing and upcoming projects. Right now, we're conducting surveys at national parks in the National Capital Region, Prince William Forest Park, Manassas Battlefield, and the C&O Canal. We're also working with the James River Association, doing mussel, fish, and macroinvertebrate assessments across the James River Basin in their riparian buffer sites. And lastly, we are starting a project this year at Quantico Marine Corps Base to do assessments for dwarf wedge mussel again. And that's it. Thank you.
Thank you so much. Next up, if we got your computer back on, Kelly Crandall.
Hi, everyone. Sorry about that. National Grid knocked on my door and was like, hey, can you lose power for 30 seconds? And I was like, right now? And they're like, yeah. And I was like, great. So I'm Kelly Crandall. I work for the New York State Department of Environmental Conservation. I work very closely with Annie Stupik, who you heard from earlier, and Amy Mahar, who introduced herself in the chat, as well as our museum folk. I work in the Western New York region, so I have a lot of our federally listed species that are located within the state. So we have northern riffle shell, rayed bean, club shell. We are starting some eDNA projects to further investigate the status of snuffbox and salamander mussel, which I'm very excited about. eDNA is still pretty new to me. And then I'm hoping to eventually maybe work on some restoration projects for those earlier species, as well as some state priorities, such as baby rayed lamp mussel, which is state threatened. So mostly work on a regional basis, do a lot of the SSAs, work on state wildlife grants, and always looking to learn. I'm still very new to the mussel world. But it's great to already be seeing some familiar names and faces. So I'm just very happy to be here.
Thank you so much, Kelly. All right, how about Ryan Heisey?
Hey, good morning, everybody. I'm Ryan Heisey, and I'm a fisheries biologist working for Duke Energy. And one of the things that I do that's related to this group is I have a set of long-term mussel surveys that I conduct on the Catawba watery system. It's for the life of our license. This is the federal energy regulatory license, so it's a good 40-year study. And so we work from the headwaters of the Catawba system all the way down into the Wateree River. So thank you, and I appreciate what everybody's doing.
Thank you so much. All right, James Schallenberger.
Hey, good morning, everybody. I'm Jamie Schallenberger, and I'm with the Susquehanna River Basin Commission. And I'd like to give a brief update and a shout out to a group that that I'm the chair for. It is the Chesapeake Bay Watershed Freshwater Mussel Partnership. We're kind of extending efforts that have existed for a long time, but our principal focus is on the watersheds that drain into the Chesapeake Bay, primarily due to the total maximum daily load activities that have been underway there for about 4 decades. We're loosely defined by the ecosystem geography, but we have members and participation from outside that region. I'm not a malacologist, but we have a steering committee that represents many folks you've heard from today, and we have a broad spectrum of people who are interested and share some kind of interest in bounds around freshwater mussels, their ecology, their potential as water quality sentinels, as well as ecosystem function. So I'll drop a link to our webpage in the chat. There's an intake form if you would like to be part of our distribution list or stay aware of things. We put information out on our website. We support folks who are seeking grant funding and perhaps need partners or letters of support, or even in some cases matching resources. And we have had a couple of virtual symposia representing our members, and we're constantly looking to network folks in our sphere in all manners. So thanks. Appreciate what's going on today, Loretta.
Thank you. Thanks so much for joining. Yeah, we'll make sure that contact information is on that contact sheet too, so anybody can reach out to you that way also. Next up, we have Mike and Leah. After that, we're at our last group. So if I have missed you, I'm so sorry, but we still do have a couple minutes. So send me another message and I will get you on that list. Otherwise, we're nearing the end of this first section. So Mike and Leah, I'll hand it to you.
Hi, thanks, Loretta. Hi, everybody. My name's Mike Demopier. I'm a mussel biologist with Cornell's Water Resources Institute, and I'll be highlighting some of our mussels projects and our team here in upstate New York, particularly in the Hudson River and the Grass Rivers, 2 Superfund sites that have seen some PCB dredging, remediation efforts, and now we are tracking kind of the recovery of these mussel resources in these water bodies. And so I'll start with the Hudson River Superfund. So if you're unfamiliar, the Hudson River is a 315-mile long river that flows south out of the Adirondack Parks to the Federal Dam in Troy, which kind of demarcates the transition from a freshwater non-tidal river system to tidal estuary before it extends 150 miles down to the New York Harbor. The Hudson River Superfund site starts kind of up in the upper reaches above that dam in Fort Edward and Hudson Falls, New York, where 2 facilities discharge for about 30 years PCBs directly into the river. And the EPA classified about 200 miles from that area downstream as a Superfund site, which kind of makes it one of the larger Superfund sites in the country, if not the largest. And where we kind of tie into this is specifically the 40-mile stretch of river from the Fort Edward plant down to the Troy Dam, where probably the most contaminants were deposited over that time frame. And it was determined to remediate this area by removing contaminated soil or sediments, and the dredging took took place between 2009 and 2015 and removed about 2.75 million cubic yards of contaminated sediments. And our mussel team, before I started here, began surveying using quantitative excavation techniques to quantify basically this the remedial activities impact on the mussel resources throughout this section of the river. And early within the first 5 years of surveying efforts, it was estimated that about 50 million freshwater mussels were destroyed from the remediation activities. And that's just kind of a little bit of the background, but we're continuing to monitor the recovery of freshwater mussels in this section with a few different partners such as DEC, the New York State Museum, USGS, and now Cornell. And we are continuing conducting quantitative excavations using a random stratified approach throughout the river. And we do have a reference pool above the PCB source. And for 2026, we are anticipating to get done 3 pools, including that reference site, as well as 2 downstream pools, Thompson Island and Northumberland. And with this data we're collecting, we're hoping to continue to track the recovery in specifically the remediated sections of these different pools within the Upper Hudson and compare, the demographics, the age structure, the densities, and the community composition to those in non-remediated areas adjacent to where the dredging occurred. And yeah, that's kind of what we have in store for 2026. I'll highlight a few of the people that have been working on this for much longer than I have, but Denise Meyer, the curator of malacology, is one of the lead co-PIs on this project since its inception, I think 14 years ago. Sean Madden, who I don't think's on the call, he was also, a lead on this, in addition to Jenna Dodge and Zoe Gliasco with the DEC, and myself. I'm the newest addition. I joined the group back in June, and I'm excited to continue working and learning more about this system. And with that, I'll pass it over to Leah Gorman with the DEC, and she'll talk more about the Grass River Superfund.
Thanks, Mike.
Leah, are you?
Yeah, I'm here.
All right.
So the Grass River is up in the way northern part of New York State, and it's a similar story to the Hudson River, but on a much smaller scale, where there was historic PCB releases, and they started remediating it. There was an estimated loss up about 1 million, over 1 million mussels, but because of our work on the Hudson River at DUC, we decided to kind of try to get ahead of that loss and start trying to recover as many mussels as possible prior to the sediment dredging and capping. Next slide. Thanks. So it's been a multi-partner and multi-stage effort, including DEC, the New York State Museum, and also the St. Regis Mohawk tribe, who couldn't join us today. We started the recovery in 2017, 2 years ahead of the remedy. And we basically were able to move over 460,000 mussels out of the way of remediation and into areas of the river that within the same 7-mile stretch that were not slated for remediation. And so they're kind of being held there now. And the St. Regis Mohawk tribe also started propagation efforts. So they've they've propagated over 5,000 mussels so far, mostly Lampsellus radiata. Um, the main part of the remediation was completed in 2021, but there were a couple years of ice scour and cap maintenance that had to happen. So the mussel work has been paused for a few years, but just kind of got up and running again this past summer. Um, so uh, Here, the next slide. Oh, I'll get to the next step, but I just wanted to highlight the species that we've found. And what's kind of neat is when they did initial mussel surveys in 2015, there were 9 species found, and um, due to the many years of just scouring and looking for mussels, we've come up with 16 species to date. 7 of those New York State designated species of greatest conservation need, and they're all in what we'd consider safe areas currently. Next slide. So our next steps are SRMT, and along with help from the U.S. Fish and Wildlife Service, are going to continue to propagate more mussels for release. The DEC and the museum are going to start this founder colony concept where we're going to source mussels from our previous release sites and basically release them into completed remedial areas to encourage the population to recover back to pre-remediation conditions. So next year, we're going to be starting to do a lot of habitat assessments and quantitative surveys to understand recruitment that's starting and try to figure out the best places for these mussels. And there's also going to be some biosubstrate placement throughout the area by Aquatic, the responsible party. Next slide. And so this is just to highlight the team. So a lot of overlap from the Hudson River, as well as the people from SRMT. I'm currently the lead at DEC, but the project wouldn't be possible without Rebecca Quayle and Corbin Gozier, who have moved on to other roles at the department. Thanks for having us.
Thank you, thank you. Thank you both for sharing. Next, we have David Campbell.
Yep. Let's see. Find the right window here. Here we go. So I'm at Gardner-Webb University in kind of southwestern North Carolina, Broad River drainage, and have projects ongoing looking some paleontology. Actually have a few freshwater snails out of the deposits early ice age. Also various molecular systematics projects. Of course, most of them teaching general courses like what I need to be to in about 10 minutes here. Do a little bit of sampling in the area, so may be able to help if you've got projects in this region. Last fall, I did just a basic invertebrate survey in the Broad River. The freshwater snails like rocky substrate, and mud from Helene is not rocky substrate. So those numbers are definitely down. We did find one live elliptio just in the area for macroinvertebrate sampling, so they are still around a little bit anyway. One paper which is sitting at the printer for the past month or so, should be out soon, is a review of what we have globally for cytochrome oxidase for mussels. So that may be of interest as it comes out. Bit of updating some of the genus classifications, some of the ones, we've known for a while that these don't all go together, but some of the name updates coming along. So that's a quick summary of what I've been doing. Thank you.
Thank you so much. All right, and I believe last but not least, we have Maddie Mueller.
Appreciate it. Hey, y'all, I'm Madison Mueller. I'm in the Biological Sciences Pathway program
with U.S. Fish and Wildlife Service. I'm in the Tennessee Ecological Field Office, so out of basin, but just a couple things that are coming. I finished my doctoral dissertation at NC State with Greg Cope last summer. So we have one publication that came out last spring. I'll drop in the chat. And hopefully just a little bit of foreshadowing. This was a conversation that we had with propagation biologists about what challenges they had and the needs that they had for assessing freshwater mussel health. So then after that, now we have one publication that is in review. It's a method development for how to assess freshwater mussel immune status just out of hemolymph, so collecting non-lethally, and then trying to make that relevant to field work. And then two more that are just about ready to be submitted. One describes the mucosal microbiome, so just being able to swab the surface of the mussel and look at the mucosal microbiome. Hopefully going to set a little bit of a framework to how we might be able to design either an intervention or a probiotic or a prebiotic that can then be used in propagation facilities to support a healthy microbiome and how we can track that over time. This was done in a controlled environment, so just at a Harrison Lake National Fish Hatchery. One of the species we used was brook floater, so we're excited to see all the interest around that species. The other one is a biomarker discovery panel, so both for metabolic and immune status biomarkers that hopefully we can kind of put together into a panel that's easy to use and again track over time. So if anybody wants to chat about mussel health, let me know. We do have a group that is starting to form just to chat about what all we can learn from hemolymph and then all of the experimental stressors that then might change those biomarkers.
All right, thank you, thank you. So that wraps up everything before lunch. So thank you all again for coming so far. We'll continue after lunch. Real quick, I'll just drop some more discussion questions just to remind everybody what we're kind of talking about. So if there's a method, tool, resource you want to share that you thought of during the presentations and haven't shared yet, that's a great time to drop it in the chat. If there's any skills, experiences that you're hoping to gain and want to link up with somebody, that's a good time to ask for the help as well. So I'll post those in there. Otherwise, we'll see y'all back here at 12:30, and I hope y'all have a great lunch. All right, hey everybody, welcome back. I'm gonna give just another minute for people to filter in here after their lunch. I hope everybody had a great lunch. I just posted in the chat again that contact sheet in case you're joining us after lunch. I know there were some folks that could only come to the second half of the day. Or if you have not yet shared your contact, please add what you're willing to share to the contact sheet and I'll send that out after the meeting. All right, so this afternoon, we're going to do some featured talks. These are talks that people had a little bit more to say than their intro couple minutes. So we're going to work through those. Let's see, I've got the list up on my screen right now. If anybody has changed their mind or has any changes that need to be made, send me a message and I will try to make those on the fly. Let's see. Otherwise, Erin, you about ready to go, or should we hold off a couple minutes?
Hey, I am just wrapping up a walk with my dog, so I would love to go a little later if that's all right. Thank you.
Totally fine. How about Lindsay? Would now work for you?
Yeah, I can go now.
That sounds great. Give me 1 second to get my screen share stopped.
Okay.
And it should be all yours. Thank you.
All right, let me find my screen share.
I feel like I have to relearn Zoom every single time I go on.
I hear you. I also, if you saw how many videos and things are open. All right, let's see if that worked. Do you
Beautiful.
see? Perfect.
I see your full PowerPoint, not just the presentation, the full thing.
I know, and I don't know why, but we're just gonna roll with it.
Totally fine. Thank you.
Yeah, of course. There we go. Okay, so again, sorry to be reiterative, but I am still Lindsay Zarecki with the Greensboro Science Center and also here representing the Association of Zoos and Aquariums. I know I mentioned earlier just briefly that we, with the AZA, have been working to create and publish our first program plan for what we call Saving Animals from Extinction, the North American Freshwater Mussel Program. And so just for a little background, in 2015, the AZA decided that they were going to take on this concept of SAFE as the flagship program for the accreditation. Every institution that is accredited through the AZA is required to conduct conservation and field conservation. And it was believed that if we come together and rally behind these published plans, that we could empower all the facilities, large and small, to contribute to conservation and support field partners that are conducting field work. So this started with a small group that has slowly grown. And when we began this endeavor, this was our team. So you can see the players that came together as our steering committee to bring this all to fruition. It has grown since then, which is one of the reasons I'm presenting this today. But in addition to these AZA facilities, we also started with a few other partners. And so these were our partners that when we first reached out and said, hey, we want to figure out who's doing mussel stuff and how we can work with you, these were the groups that replied to that response. Obviously, there's a lot more people out there doing work in freshwater systems and with freshwater mussels, but when we were just getting started, this is where it began. A requirement to conduct a program plan through this SAFE is that whatever you produce, whatever goals, objectives, etc., that come out of your program plan, they are required to be tied to an established recovery plan. When this group started, there was a lot of conversation about, do we aim to focus on a species or a grouping, or do we just go with all mussels? And what was decided was that we would follow the national strategy that the FMCS put out and that we would just target freshwater mussels. Now, AZA does expand beyond our country. We do have partners in Canada and some that are in Mexico and South America. And so we opted to just focus all of our work on the North American freshwater mussels. What came of this was we did a 2-day workshop to put together our plan. And then out of that plan, which included members from FMCS on this, we published our vision. So our vision became that AZA North American Freshwater Mussel SAFE envisions a world where native freshwater mussels and their habitats are thriving and people appreciate their contribution to healthy aquatic ecosystems. From there, we realized that AZA facilities, we want them to collaborate with partners in freshwater mussel conservation through areas such as outreach, education, research, as well as propagation. Many of these things are already happening, but we know that they can be happening on a larger scale. It's just a matter of figuring out how we do that. So after that meeting, we tied our goals to that recovery plan. We went through various versions of goals, and these are the 4 that we've come out with for our inaugural program plan. It is a 3-year program plan, at which point we will revisit and we will be required to draft a subsequent 5-year program plan. What we wanted to do was to create a variety of options where institutions, AZA and otherwise, can get involved as they would like. Institutions that do get involved in the program do not have to be actively contributing to every goal. They just need to figure out what works for them. And then they can form or they can join subcommittees and they can start to participate with our groups. And so we went to the AZA conference, which meets twice a year. There's an annual and a mid-year. We went with stickers, which here's a representation of our stickers that we handed out. And we found that there was a lot of interest from the zoos and aquariums in our country to get involved in freshwater mussel conservation. That ranged from having better outreach and education to actually propagating mussels on site, to holding animals, and to supporting other agencies and organizations in their community. We learned pretty quickly that a lot of facilities have some type of freshwater on their property or next to it, whether it's a stream or a lake or a pond or whatever. The capacity to do more is definitely there. And it was really rewarding to see that the interest to do more was also there. So the goals that we have come up with, our first one is to really help what was at the time FMCS, but really all of the mussel biologists out there to understand stakeholder analysis and engagement. It's funny, you come to these meetings and you see faces and names, and you're like, oh yeah, I know that person. But at the same time, we all tend to go back and we do our thing, and it's a matter of who's doing what around us, not just here, but all across our country. And are there ways that we can be supporting each other? We have found that there was a lot of interest in having, at least within AZA and the handful of partners that participated in our study, there's interest in having education and outreach that has a consistent and inviting vibe to it that we could all buy into. Of course, everybody was interested in financial support in some capacity. Who doesn't need money? There was interest in doing more for habitat restoration, and this really derived from another SAFE program that's out there that I'll talk about in a moment, and how well they're doing when it comes to promoting habitat restoration, and can we do that same concept but for freshwater mussels? A lot of institutions were interested in getting in the field and doing monitoring and helping others. Surprisingly, many institutions already are out in the field helping their partners to do surveys. Mostly our aquariums, but some of our zoos were also interested in being able to host fish or to host mussels. We have a lot of aquariums who are really good at growing fish. That's what they do. And so there was conversation of, does it make sense to work with our aquariums and have more host fish available? I know many of us, when we need the host fish, we are actually having to go out in the field and find them. And to be able to have a repository of host fish would be lovely. I know I would love that for our propagation facility. And then one that I'm not as familiar with, but some of our institutions are, was advocacy. We do have government affairs groups within the zoo field, and they are very, very talented and strong at advocacy. And so we thought that was another area that we could actually help with, help our stakeholders with the level of engagement in mussel conservation. Our second goal was to build assessment capacity, and this one is something we are already working on. So we are tied in with the IUCN, who is doing updating of their red list for mollusks. And this group, the Safe Freshwater Mussels, has taken on updating 143 freshwater mussel species red list. The snails are going to somebody else, but the freshwater mussels are coming to this group. There will be a workshop in May hosted through the Center for Biological Diversity at the Indianapolis Zoo to review the red list reports, to get everything updated and concise. We have 6 master's students who are all working in the GIS world to help update all of the maps associated with these species. And ultimately, we can contribute this back to the IUCN because, as we know, people don't protect what they don't know about. And so we've got to get the information out there. So we thought this was a great way for our group to contribute to mussel conservation globally. Another one is the conserve habitat and protect wild populations. As I said just a moment ago, a lot of facilities have freshwater bodies on their property, from streams all the way up to lakes. Granted, some of our aquariums are on oceans, but a lot of them have freshwater. What we have come to realize is that those freshwater habitats present themselves as a beautiful exhibition that we can be using to tell a story. We always talk about habitats that the animals we present live in, and conserving those habitats. Why not promote that same story with a habitat that's right here in our backyard? What we have found is that Safe Monarch Butterfly has done a really good job at this. They have a beautiful standardized statements on how to put in pollinator gardens and ways to protect pollinators, from simple things to really grandiose things. But a lot of the concepts that they're using, we realized were applicable. Things like the lack of chemicals in an environment and the not using pesticides and having healthy native vegetation in this space, the importance of a riparian zone. You guys all know this, but what we realize, our general public and visiting audience does not, but it is the backyard that they live in. Therefore, they are innately engaged in wanting to save it. And so we want to work on creating this healthy habitat program. In tandem with that, our fourth goal is to overall increase our education and awareness. And we have a subcommittee of educators from within the zoo field who are tackling this endeavor. So they are looking to improve education and outreach within our AZA community, and we're happy to extend that beyond our AZA community, and to get our arms really wrapped around the public perception of freshwater mussels. As I mentioned, we have a propagation lab at the Greensboro Science Center, and people come by and they talk to us. The number one question we get is, can you eat them? And in all of the work that we're promoting, and we have graphics and signs and videos, and they can see mussels, and the first question is, can I eat it? We realize we have some work to do. We want people to want to protect these animals, not as a food source, but as an ecosystem builder and engineer, and something that we need to have in our natural environment. So this group has their work cut out for them, because freshwater mussels are not a species that people know a whole lot about. So that is our SAFE program. We have two co-leads, which is Monty Bohm out of the Indianapolis Zoo, and then Jacob Harmon, who's an aquarist at the Mississippi River Museum and Aquarium in Dubuque. And then I am the liaison for this group. I liaise between them and the AZA as a representative of the Wildlife Committee. So I wanted to present this today because this is something we've been working on for multiple years. And what we're trying to do is just get it launched. It wasn't produced until December of 25. It's really new. But now we want to reach out into the freshwater mussel community and learn more about who's doing what and where could you use the help. And as you think about that, are there ways that institutions like zoos and aquariums can help you? Whether it's manpower, it's messaging, variety of different ways that we can work together. And like I said, every AZA institution is required to participate in conservation. So they're doing a lot more than just outreach and educating. They can get in the field, they can help support you in your endeavors. So it's a little bit of a non-traditional presentation for a group like this, and I recognize that. But we see millions of people when it comes to zoos and aquariums, so we have a captive audience that we would love to inform and bring awareness to and empower them to protect mussels in our backyard. So thank you guys for letting me talk on this. I am happy to answer any questions. I can also place in the chat a link to the published plan so you can see what we have put out there. But ultimately, we are here to support freshwater mussel conservation and to figure out how AZA can best fit into that bigger picture.
Thank you so much, Lindsay. We do have time between these afternoon talks. I planned in a little time for questions and discussions. So if anybody has anything for Lindsay, we can do that now if you've got anything. Give you a second to answer. If not, feel free to carry on in the chat. Always good. I don't think I'm seeing anybody pop up, so we'll carry on to Erin, please.
Awesome. Thank you. Hi, everyone. Are you seeing the big main slide?
Yep, looks good.
Fantastic. So I'm here to talk to you today about maybe something you've heard of. I've heard a couple of folks mention their engagement with the Mussel Assemblage Health Index, and maybe this will be new to a few of y'all. So want to start with, of course, huge shout out to so many people that have been working on this, but specifically to Wendell and Tracy, Angela, Steve, and so many others that have really been the backbone of this work. And I get to play the role of engaging a little bit more in policy and rolling this out. So let me see. So first, let me tell you a little bit about American Rivers. So if y'all weren't here for my intro earlier, I'm Erin McComb, Southeast Conservation Director with American Rivers. We are headquartered in DC, but we have field offices across the country. I sit in Asheville, North Carolina. And our vision is probably something that resonates with a lot of y'all, is a nation where every river is clean and healthy for people and wildlife. So first, I'd like to start by telling you a little bit about me and some of the places that have informed who I am. So I grew up on one of the hardest working rivers in America, the Mississippi River in Memphis, Tennessee, but I fell in love with rivers on family trips to the Spring River in Hardy, Arkansas. And I would don my goggles and swim with the silvery shiners below a roaring drop in the river for as long as I could pretend not to hear my parents telling me it was time to go. Later at Appalachian State, my master's work revealed the world of freshwater mussels to me, and I discovered how creative and cryptic they are, and how they're better anglers than any I know. So I put on my mask, now upgraded with a snorkel, and I would survey streams in almost every county in Alabama, from the clear, swift streams to the slow, hot, and stinky ones that I still regret getting in. And so it was a part of that experience that I joined you, the community of freshwater mussel folk, and I learned of the daily grief that goes along with this work. We're losing these filter feeders. And people who don't know much about mussels at all still benefit from cheaper, cleaner drinking water. And the people who do know about mussels, like us, know that we don't want to know what a world without mussels will be like. And that brings me to today and the work I get to do about mussel decline with an absolutely incredible group of partners. So I'm going to play this short little film that'll do a good job and give you some fun images to kick off this work that I'm going to tell you about.
Since the 70s, freshwater mussel populations have been declining, and we don't exactly know why. Over 70% of freshwater mussels are at risk of extinction, and they're the most imperiled group of organisms. They're going extinct at faster rates than any land or ocean species. At American Rivers, we believe that biodiversity protection is really important, so we're working in partnership with folks across 13 states to investigate what's really going on in the rivers and streams of the Southeast.
This part of the U.S. has more mussel species than anywhere else on Earth. Mussels appear to be integral components of stream ecosystems. As filter feeders, they're really intimately involved in the cycling of nutrients. They process all the material that's coming down a stream. They take a lot of that stuff out of the water and they make it available to other organisms.
Freshwater mussels can filter up to 30 gallons of water a day. They keep the rivers and streams really clean, which is important for healthy functioning ecosystems and is important for people to have clean, safe, reliable drinking water. Rivers are the most threatened ecosystem on the planet. This research will help us inform smart science-based policy and practice to ensure that biodiversity is protected in the future. It's important for us to understand these root causes so we can craft policy and practice to address this decline before it's too late.
All right. So you might see some of your organizations on that list. Thank you so much. All right, so why does basic mussel health matter? The aim today is to discuss why it matters and why existing tools don't address our problems, and how the Mussel Assemblage Health Index can help and why it's valuable for agencies. So as we established, mussels aren't doing great. The example graph on the top right is Horselet Creek in Kentucky, where mussel abundance declined over 98% in 15 years. And declines are studied in limited context, and large-scale patterns are not studied or causes not known. Existing surveys and data are hard to compare across basins or states because we lack a standardized health index. So we set out to change this, and the map shows a lot of the study areas that we investigated. So shout out to all the agency folks on the call. Agencies collect excellent mussel data, but existing surveys and data are hard to compare across basins or states because we lack a standardized health index. They rely on untested assumptions such as which species are tolerant and subjective expert opinion as well. As a result, two biologists might look at the same river and reach very different conclusions about its condition. So past tools like MCAT, MCI, MIBI, SSQI are region specific and often developed for the Midwest or single rivers. And since agencies already collect excellent mussel data, the Mussel Assemblage Health Index turns that into an objective, repeatable measure of ecosystem health. So this was developed by Wendell at the U.S. Forest Service and many partners using data from 50 streams across the eastern U.S. and Canada. This was published in Freshwater Science in 2014. And it's designed to be objective, so free from bias, broadly applicable, works from Appalachia to the Midwest, and practical, uses standard survey data. So at that link there, americanrivers.org/MAHI, you can find the free Excel-based calculator. I'll share those links in the chat at the end as well. So as I already said, Mahi is applicable in streams across eastern North America, calculated on commonly collected survey data, flexible, can use different kinds of data, is objective and free of untested assumptions. So Mahi combines 4 equally weighted indicators, each scored 0 to 10. So the first is the proportion of historical richness present, which measures species loss over time. The second one is assemblage recruitment strength, which measures reproduction and replacement. Third, density relative to stream size. So that's abundance standardized for watershed area. Fourth, population growth. So trends in density over time. And so together, these represent persistence, reproduction, abundance, and trajectory. You might remember this slide from earlier. So Mahi clearly separates healthy, a median score of 8.3, and degraded there, that median score is 3.2 assemblages. Results closely match expert judgment, but remove bias. I think one thing that is particularly interesting is that it's robust to missing data. We know that we don't live in a perfect world, and there's maybe something that's always missing. So the score changes less than 1 point if one metric is missing. But it's sensitive enough to detect subtle declines or recovery. So it can, as a person who spends a lot of time doing actual restoration work, this is ideal for tracking management success, which is really important. So the slides that I hid to get into the time really got into data attached to each one of these, but mussel declines can't be attributed consistently to general stream degradation. And I think the cause of enigmatic mussel declines is likely uniquely affecting mussels and isn't caught when states are conducting Clean Water Act assessments. So I had a few slides that were kind of comparing this to fish and bug IBI scores and other things used under the Clean Water Act for biological integrity. So this looks at Mahi evaluation and performance. So it's comparing expert scores. And so it eliminates individual subjectivity, but was still really in line with expert opinion. Again, insensitive to missing metrics. So the vertical axis is looking at difference in index score, and you can see what happens if any of those individual metrics were missing. All right. Altogether, muscle declines are often not associated with typical forms of stream impairment. Index of biological integrity scores don't reflect the health of muscle assemblages, but fish and macroinvertebrate IBI scores don't always align either. So fish, invertebrates, and mussels often seem to respond differently to stressors. So we argue that incorporating mahi into a bioassessment makes it more robust. All right, so why might this be valuable to you working at an agency? So it's a single, easy to interpret number. It can help detect declines early and provides quantitative evidence for restoration outcomes. It can support reporting for Section 6, state wildlife action plans, and funder metrics, and enables regional coordination through a shared data standard. So a couple of example applications, you might track recovery of mussels after a dam removal. This is the Ward Mill Dam removal on the Watauga River, or another barrier removal you might use it for. You could measure biological response to invasive species like Corbicula or Dracaena, evaluate outcomes of flow or restoration projects. Michael maybe will stop a pump storage hydro peaking operation, and we could use it to look at that. Report baseline river health and public and policy communications. So the call to action, we'd love it if you could pilot mahi in key watersheds to establish reference conditions. Use your existing data, like I know folks in Florida have done. Shout out, thank you for that work, to calculate baseline scores and identify trends. Collaborate with us, with the Forest Service, American Rivers, and partners for support, and integrate mahi into monitoring frameworks and grant metrics. All right, in closing, adopting mahi helps unify freshwater conservation and management. It provides a common science-based language for describing mussel assemblage health, transforms years of agency data into actionable insights, and builds regional consistency, early warning capacity, and measurable outcomes. So needs and next steps. So what's on the horizon? Wendell and I are going to put together a series of trainings on the Mussel Assemblage Health Index that will include some case studies. So it'll get into the nitty gritty of the tool use, but then hear from folks that have worked with this. So if any of you have played around with mahi and ran into some challenges or had a really good time with that, would love for you to let me know about that. You can scan that QR code there. It'll take you to a Google form, and you can be kept in the loop about when we'll be giving those trainings. And I imagine that we'll have 2 or 3 of those, and we can record those so they can be on a website. But we want to keep track of the folks that are doing this and seek to expand adoption. We'd like to adopt mahi as a standard tool for assessing stream health at the federal and state level to ensure healthy rivers. You'll see this QR code. It's all the same one. It's just taking you to that Google form if you want me to let you know about updates on mahi and trainings and things like that. But this is what the Excel app looks like. Another shot there. So right now, this is American Rivers regional offices on the map there, and a couple of the states that we're initially targeting for adoption are Minnesota, Kentucky, and North Carolina. But we would love to expand it obviously beyond that. And that's what I've got. So there's my contact information. Feel free to get in touch. Feel free to put your info in that sheet, and would love to stay in touch about this. So thanks again for organizing this meeting, and appreciate it.
Yeah, thanks so much. Thanks, Erin. Does anybody have any questions for Erin? Alrighty, I don't think I'm seeing anything. But there's a couple in the chats popping up. just if you could maybe drop that QR code in there or a link, that would be amazing.
I will do that. Absolutely.
Thank you. Thank you. Alrighty. If that is it for Aaron, we will head on to Alex. Thank you.
Alrighty. Can everyone hear me again?
Yep, you're good.
Okay, perfect. Let me share the screen. Share. That should be working.
Yep, we've got your first slide.
Perfect. Alrighty. So, it's not giving me presenter view for some reason on my side. There we go. Okay. Sorry while I get this working here.
No worries. The struggle is real. I was fussing with presenter view this morning too.
Oh goodness, yeah. And just before I signed in, my computer said my internet connection is unstable. So just fantastic news. But yeah, so again, my name's Alex Crawford. I'm the Canadian in the group. I think the only one, if I remember. Um, and I'm doing my master's at University of New Brunswick. Here's my super long title, Advancing Freshwater Mussel Monitoring. So I'm doing a comparative study of survey method efficiency and morphometric identification accuracy, and that's with Dr. Michelle Gray in her lab. So an introduction to the freshwater mussels that's not really relevant to this group. Everyone knows how important they are and why we use them. This is just part of my spiel when I present to other students who don't know a lot about them. And to be honest, the freshwater mussel community is really, really quite tight knit. Down here on the East Coast, I only know of two other labs and other universities that work on it. Otherwise, it's pretty much just a handful of us that care and try to reach out to a lot more organizations in the US. As we know from this meeting, there's a lot more work happening down there. So I will skip to the research that I was working on. So my first objective was to compare the standard methodologies, being visual surveys, so just snorkel and mask, like we've seen here. And the other one is a tactile. So this was a method that was used a lot in the lab. I didn't find it in literature per se. That's not to say that it's not used, but that's just going along and inserting your fingers into the substrate to try to find buried mussels. Now, most people do that as at the same time as the visual type of method, but I wanted to assess what mussels we were finding based on each method to try to see if one was more efficient than the other for finding specific species and what have you. And the objective two is a morphology-based species classification. So I wanted to look at if it's possible to correctly identify or at least have an estimation error mussel species based solely on their external characteristics. So if we can't do lethal sampling or get a DNA sample more for citizen science type outreach stuff, because this picture here is on Grand Lake, and it's a very popular lake for recreation, boating, swimming, what have you. And there's a lot of citizens that live on the lake, because I live on it as well, that come to me and ask all the time, what is this? What am I looking at? Yada, yada, yada. So I was looking to see if I can make that a little bit more accessible and easier to the public. So my study site selection, I used LiDAR downloaded from a source here called GeoNB. The entire province has been mapped with LiDAR and is freely accessible to us, so that's really awesome. So I was able to download the tiles and try to estimate where sandy depositional areas might be. From that, I chose 67 sites that I wanted to go to around the lake. And then we surveyed them, the team and I, and we would note them as ideal, not ideal, or not suitable. hoping to find yellow lamp mussel as kind of the background goal, but not the pure goal. Just, hey, it would be really nice if we found them, so let's limit or try to focus our sites that way. So an ideal site would have been limited vegetation, sand-dominant substrate, and presence of either live mussels or mussel shells. Not ideal was potentially viable, but not currently optimal. So meaning next year or the year after it might be okay, but right now it's not. Grand Lake is massive and sometimes can behave like an ocean. We get 3 to 4 foot swells. So something that's not good this year will be entirely different next year. The coastline changes year to year, not macroscopically, but in terms of microhabitats that freshwater mussels use, big differences there. So um, this map was from my 2024 field season, and then I revisited all the sites again this past season, 2025, and sure enough, the sites were very different when I returned. And then not suitable, they were noted for things that aren't going to change, so rocky substrates, really heavy rooted vegetation, and little evidence of mussel presence. So we noted those as not worth surveying. Something else I was looking into on this project was some modern survey methods. So the use of aerial drones and underwater ROV. So the goal was to use these and evaluate how they would be used for habitat conditions. They were never intended to help identify mussel species. As we all know, the uh, aerial and ROV technology just isn't there yet. But the aerial drone was really good for habitat context. So when I was trying to map out my sites using LiDAR and Google Images and what have you, satellite imagery, I was able to get an idea of the landscape for sure, but the real-time microhabitat, as I was saying, from Grand Lake changes sometimes day to day. So being able to fly up the drone and get a good visual of the sandbars or potentially any other little like freshwater input was really helpful day of, so we could adjust our survey methods or at least the survey site reach on day of. And then the underwater ROV survey that we were intending, the lab bought this hoping that it could identify mussels that we could get a stable enough image, but this particular model wasn't able to. We were able to get good visualization of the habitat, but not clear enough video to try and identify species. We could get overall abundance, but not species level identification. There's no onboard gyroscope, so keeping it level was very difficult. As is, the video is pretty nauseating to watch afterwards. So you have to take still images. So, but we tried it and that's what we learned. So trial and error, right? So each survey, we did timed surveys, and I apologize, this is from my field notes for each site. So Hayden and Hayden's our field tech, we got into the water at the GPS points from the previous map I showed, and then we would survey to the left and to the right for three sweeps per method at 22 minutes for each sweep. So that would equal 4.5 person hours per site. And then direction from the middle point for visual or tactile was decided randomly previously to avoid bias. And distance along the shoreline was limited to a maximum of 100 meters to either side. And some sites couldn't allow for the 100 meters due to barriers in the habitat, rocks, or large debris. So we did the timing of the 4.5 person hours to mitigate that. And then I'll just try to explain here what's happening. So can everyone see my mouse? Can you see the mouse cursor on that? Yeah, okay, perfect. So in the middle, this is the GPS line, and this line here would represent the shoreline or the bank. So the points are myself and Hayden. We would start and we would survey down and then back. Once the time stopped, that's where we finished. So then on this would be one survey sweep. Then this one would be, okay, so we made it this far and we still had time left. Then we changed our sweep pattern to try to cover more ground. And then same with here, we would go up and then we would only try to survey the areas that we already saw. So we try to stop at the same point from the first point and then go back just to cover a lot of ground. So the visual ones, as you can see, we often got to cover the ground a lot more effectively than tactile, because tactile took a long time. No matter if you were finding mussels or not, it wasn't really slowing you down if you were being efficient or effective, sorry. So these are just two different site examples. This first one here on the left, the substrate was a little bit more rocky, so tactile was a little bit more difficult. And then on the right here, we had a sandier substrate, so we were able to cover a bit more ground. Next, there we go. So yeah, the traditional survey methods I was looking at were visual. So just myself in a wetsuit and a snorkel, as you can see, having an absolutely great time compared to some of my other graduate students. So that is pretty straightforward. And then the tactile survey involves going along, limiting, so keeping your head above the water to limit your visual senses, and just inserting your fingers to the depth of the second knuckle for me, which was 6 centimeters. So we just went with the second knuckle to keep it consistent. And the survey procedure, so we collected the mussels with dive bags, and then we collected their measurements using calipers, height, length, and width. I wanted to make sure I noted what I was calling height and what I was calling width. Length is pretty consistent in literature, but height and width tends to get flip-flop between, so I just made sure that I shared what I was calling height and what I was calling width. So yeah, here's a little result just from the underwater ROV survey. So like I was saying before, it's good for just getting an overall image, potentially even some photos of potential fish host species, which can help indicate what mussels might be there. But we can only use the still images. The video is very shaky, but when we do get a few good still images, we can see clearly just overall mussel presence. But if you try to get close in this above image, you see that the impellers on the drone or ROV itself actually kicks up the substrate. So it kind of is its own worst enemy in that way. It's also really susceptible to water current and water movement. So any kind of tide activity or heavy waves, and it just absolutely gets thrown around. But yes, again, useful in noting fish host species. And yeah, that's pretty much what I've been working on so far. I don't have concrete results to share yet. That'll be coming out hopefully in the coming months here as I write my thesis, but thanks for listening.
Thank you so much, Alex, and thanks for being here and representing Canada. It's great to have you. Does anybody have any questions for him? All right, I don't think I see any popping up. Feel free to speak up if I missed you. Otherwise, we'll head over to Stephanie.
Thanks so much. Let me just get my slides going. Okay, and hopefully you're seeing my full PowerPoint screen. Sounds like yes.
Yep, we gotcha.
Perfect. Okay, hello everyone. I'm Stephanie Farrington. I am a PhD student, recently defended last month in Alison Roy's lab at the University of Massachusetts Amherst. And I'm also a pathways biologist with the U.S. Fish and Wildlife Service. So I'm going to share one of the projects that was part of my dissertation work, still actively revising that, and there'll be a little call to action at the end there. But this project is focused on identifying ecologically relevant host fishes across the Atlantic slope. So I don't need to explain to this group why freshwater mussel host fishes are important and interesting, but just to kind of set the framework for how I'm thinking about them. Understanding these mussel host fish relationships can help us to inform population status. So like the long-term viability of an area, we know there's not going to be reproduction and recruitment if there aren't access to suitable host fishes in an area. Can also help us to inform areas that are suitable for interventions like population augmentation or stocking. And it can also identify areas that might need habitat restoration initiatives to improve fish passage, so those mussels are able to access their hosts. These relationships can also help us to inform species distribution models, which can help us think about species native ranges, as well as potential routes of dispersal for these animals between watersheds. And then, of course, host fishes are really important for mussel propagation. We can think about them in terms of species that are really good at producing lots of mussels, as well as potentially mimicking natural conditions, access to certain species or certain genetic diversity. But with kind of all of these important themes in mind, I would argue that to understand host fishes, we need more evidence than just laboratory trials. So lab trials do establish that physiological compatibility between species, but they don't really tell us anything about what's actually happening in the wild. This figure comes from a recent paper by Hopper et al. Using data from the Freshwater Mussel host database. And you can see in the graph in pink there, the vast majority of evidence for host fish relationships come from lab trials. But using natural infection evidence, so mussel larvae, Glochidia, found on fish in the wild, inherently provides evidence of sympatry, these species occurring at the same time, as well as this species interaction, which is especially important for species with specific host attraction strategies that require more host interaction. So that brings me to my research question for this project. I'm looking at, does natural mussel host fish use vary across the Atlantic slope? And to examine this, we collected wild fish and identified the Glochidia attached to their gills. So this is our study area. We're looking at the Northeast Atlantic slope here. So we sampled in 6 HUC6 watersheds, which are indicated in blue there. We partnered with state agencies that were already collecting fish for their own population assessments. So we kind of opportunistically collected our species of interest as they were collecting them. So those agencies in the Northeast were Mass Wildlife, the Maine Department of Inland Fisheries and Wildlife, and then Holyoke Gas and Electric at the Holyoke Dam. And in the Southeast, the North Carolina Wildlife Resources Commission and South Carolina DNR. In total, we collected 12 species of fish from the Persid, Maronid, and Centrarchid families. Briefly going through some of our methods. So these fish were collected in a myriad of ways depending on the agency doing the collection. Fish gills were examined in the field when possible, identified if there were potentially Glochidia present. Any infected fish were retained for dissection and Glochidia extraction in the lab. And those were collected using dissecting probes under the microscope. And then each Glochidium was individually photographed so we could do morphometric assessments. I'll briefly walk you through those as well. So we collected shell measurements, so height, length, and hinge length, as well as other characteristics like overall shell shape, color, presence of a hook. To have a reference point, we also compiled existing Glochidia measurements and characteristics from the literature. And then we supplemented those with some species that were missing from those references. Those were provided by the Maryland DNR and the U.S. Fish and Wildlife Service. And then we compared our unknown samples to our known reference samples using principal component analysis. So here's an example of what that looks like. So the light gray are going to be the known reference samples, and then the black points are our unknown samples. And here they're varying in shape by the sampled watershed. And so each ellipse here is showing a different candidate species. So again, we're comparing our unknown to the known. We used Glochidia that overlapped with these 95% confidence ellipses as the candidate species for those samples. We did have a few samples that overlapped with multiple species, and I'll talk a little bit about that in a moment. But here are our overall results. So we performed this analysis separately by watershed so that we only included native species for the sampled watershed. And in total, we found matches for 42 of the Glaucidia that were sampled from fish in 3 of our sampled watersheds. So that's the lower Connecticut River and the Penobscot River watershed, and those are both in the northeast, and then the upper PD River in the southeast. The next couple of figures here, I'm just pointing out at the top there is our candidate species, and then there's an example of what those Glaucidia actually looked like on the fish. And so in total here, we identified 4 species and 1 genus that were collected within their known native range, as well as a few, the Cyclonias and the Epioblasma, were not native to the sampled watershed, but are known to exist in an adjacent watershed. Just to highlight some of those interesting mussel-host relationships that we identified. So here I'm showing the mussel at the top, fish at the bottom, and then the little sparkle there indicates whether that's novel evidence for a new mussel-host relationship, new completely to the literature, or new natural infection evidence indicated in white where there had been a previous lab trial. So starting here with Sagittunion acutus on striped bass, that was a novel relationship. We found that in the northeast. Yellow lamp mussel, Lampsillus curiosa, we found on Moroni americana and Percophlebescens, so the identified primary host for the species. We also found Cyclonias tuberculata on Lepomis auratus. These species names trip me up. But this was another novel mussel-host relationship. And then we also found Epioblasma capsiformis on both white bass and largemouth bass. And again, those are both some novel relationships. So hopefully here, this is highlighting how these host fishes can be great at sampling species that maybe are existing slightly out of their known watersheds, as well as identifying some relationships that previously had not been tested in laboratory trials. I mentioned that there were a few samples that were identified for multiple candidate species. So this is where their shell measurements and shape overlapped with multiple of these candidate species. So that happened for 4 Glaucidia that were all collected from striped bass in the Connecticut River watershed. And so for all of these samples, the candidates were either Sagittunion acutus, eastern pond mussel, and then either Lampsillus curiosa or Lampsillus radiata. And so I'll just highlight here, you can see where those samples overlap between those different ellipses. And then again, showing here, depending on which of these relationships end up panning out, this would either be a novel relationship with eastern pond mussel, or would be new evidence of a natural infection for either eastern lamp mussel or yellow lamp mussel. And so both of those Lampsillus species have already been verified in lab trials, but previously had no natural infection evidence. So kind of wrapping things up, thinking about geographic variation in host fish use. So we did not identify any mussel species in multiple watersheds, but one relationship I do want to highlight is white perch and yellow lamp mussel. So we did confirm this relationship in the northeast. This is known to be a primary host for the species. We collected a similar number of white perches in the northeast and in the southeast, but we didn't identify any Glaucidia on the fish collected in the southeast. So we can't, confirm that they're not using this as a host there, but it does sort of bring up the question for me of if white perch is considered a primary host for this mussel, but all of the evidence from this study and from previous studies is all coming from the northeast region, can we truly call this the primary host, do we need additional information from other regions, other watersheds to kind of confirm that this relationship is consistent across the species range? And so just leaving with some final thoughts here, and I'd love to hear your questions and suggestions as well. But highlighting that using natural infection of wild fishes, we provided novel evidence for 3 mussel-host relationships with no previous evidence, as well as confirming 2 known relationships for yellow lamp mussel. I also want to highlight that it's this pair of natural infection and the lab transformation really confirms that this is both possible and happening. And so some of these novel relationships need that evidence of lab transformation to confirm that this is a suitable host. A couple of next steps for this project. Morphometrics can be a useful tool for species ID, but there were lots of samples that we weren't able to identify. Looking for additional reference samples. So if you or someone you know is working with mussel Glaucidia or has access to measurements or would be willing to collect Glaucidia, please reach out to me. I would love to talk with you about some species that I'm looking for. We also collected DNA from our Glaucidia samples, and so we're working on sequencing those to validate some of these species assignments from the morphometrics. And with that, I'm happy to take any questions, and my email's up at the top there. Thanks.
Thank you so much, Stephanie. I'd actually shoot me a message later so I remember, but I've got morphometrics for most things that come through our in vitro lab, so happy to share them with you.
Yep. Okay, thanks.
Yeah, anybody else have questions or comments for Stephanie? Okie dokie. We'll head on over to Jill next then. Thank you.
Hi, good afternoon, everybody. I'm going to pull up my slides here. Can everyone see this okay?
Yep, we've got your full screen presentation.
All right, awesome. Well, like I said, good afternoon. My name is Jill Federick. I'm a PhD candidate at the University of Maine. And today I'm going to be talking to you about museum models and murals or the conservation of the yellow lamp mussel. Today, my talk is going to be like a really broad view of my dissertation, going a little bit over my 4 chapters as I'm gearing up to defend this August. So thank you so much for inviting us to the symposium, and let's get started. So I wanted to start off with talking about who is the yellow lamp mussel, what is the yellow lamp mussel, but as a lot of us have experienced this species already, I'm sure I don't have to go into too deep of detail. But the yellow lamp mussel, or Lampsillus cariosa, is a medium-sized Lampsillus species. It's bright yellow and it's got some green rays on its posterior slope. What's interesting about this species is that its status is really variable from state to state and province to province, ranging from apparently secure to presumed extirpated across its range. So disentangling why its status is so variable is incredibly important to its conservation and one of the main goals of my dissertation. Furthermore, yellow lamp mussel, as Stephanie has just gone over, has a variety of host fish. Disentangling the strengths of those relationships and how they might vary across space is also a lingering question that I aim to kind of get at a little bit with my dissertation. And finally, its habitat use can range from lentic-like conditions, impounded areas, to fast-moving water and riffles in large substrate. So figuring out what habitat is suitable, what host fishes are suitable, might help us understand why the status is so variable from state to state. So, okay, my dissertation has 4 main objectives, which are going to be colored and symbolized using these icons below. Primarily, I started off assessing the historical distribution of yellow lamp mussel. So where was it across the landscape? Then I moved into looking at modern day data from agencies to assess its range-wide habitat suitability and patterns of habitat and host preferences using species distribution models. I then kind of zoomed in at a local scale and thought about how to improve substrate and bed texture habitat analysis and the effects of dam removal on yellow lamp mussel in Penobscot County, Maine, where I'm based out of. And finally, I aim to engage and inform the local community about mussels. And I apologize because this talk today is going to be a little bit about like drinking out of a fire hose as I go over all of these objectives in scant detail. But if any of these interest you, I'll be available for questions and to contact afterwards. So when I started my PhD and I knew I wanted to assess the conservation of yellow lamp mussel at a range-wide scale, I started by pulling museum data. Museum data, as I'm sure a lot of you know, is really incredible source of information. It is easily available thanks to tools like Mussel Mapper, and it is wide-ranging, it is free to access, and you could get a lot of it with just a few clicks of a button. So, beginning my dissertation, I pulled all this museum data from organizations like Mussel Mapper and some museums in Canada. And what I did was I overlaid that to where we could expect yellow lit mussel to be, which here is symbolized by these gray polygons. This expected range I also built from Mussel Mapper, and I added some regions that we could expect yellow lit mussels, such as the St. Lawrence drainage in New York and some regions in New Brunswick and Nova Scotia. And what I found was that out of all of these mussel records, 36 of them occurred outside of this expected range. And I was wondering, why is that? Maybe they're erroneous, maybe they are in fact yellow lamp mussel. So I dug into each of those interesting museum records in turn to figure out what was leading to their persistence. And what I found was that out of those 36 out of range records, 18 of them were genuine Lampsillus cariosa or yellow lamp mussel. Those are symbolized by these bright yellow dots here, which occurred in the provinces of Ontario and Quebec and areas of Western New York and one in Vermont. And what's really interesting is that Ontario and Quebec, these two provinces have no protections in place for this at-risk species. So we published these results just in the last year, and we hope that this might help survey and monitoring efforts in these areas. We also examined why this animal might have been in Western New York, at least historically, and we found that the museum records in the occupied watersheds traced neatly the Erie Canal, which might be a possible hypothesis for why this animal could have been in Western New York. And we could disentangle some of these relationships with genetic work, which I hope Paul Stanley's MS will help us do. And I'm really excited to hear the results of that work. Moving on, I wanted to get at the current habitat and host suitability of yellow lamp mussel through species distribution models. And I'm sure a lot of us here have used species distribution models in our work, but I'm going to present the overall framework of what a species distribution model is so that we're all on the same page. So how a species distribution model works is you have the occurrence of an interesting or focal species across the landscape, right? So from survey efforts, you know that yellow lamp mussel occurs in these particular locations, and you can overlay that information with environmental data. So things like pH, temperature, things like that. And what you could do is you could use that environmental data and where the animal is found to predict its presence. And you can then use those relationships to predict across the landscape and make a map for where this animal might be likely to be present or likely to be absent, or where habitat might be likely suitable or likely unsuitable. And these results can be really important for management because instead of surveying blindly for a species, you could target areas where, okay, based on these models, based on the temperature and suitable pH, I know yellow lamp mussel is really, really likely to be in this spot. But, you know, the environment isn't the only thing that dictates where a species can be found, particularly for freshwater mussels, they're parasitic, right? So I took on what's called a stacked species distribution model approach, where you could take the occurrence data of not only your focal species, right, in this case, the yellow lamp mussel, but potential host fishes and co-occurring mussels. And then you could run a species distribution model for each species in turn, and then regress the probability of your focal species along not only the environment, but these predicted distributions of these other species. So this is called a stacked SDM. And I'm actually going to go more in depth on this topic in this chapter in the Yellow Lamp Mussel Working Group on Friday. So if you're interested to know more, you could catch us at 10 a.m. on Friday. The results of these types of models can help us, again, identify areas to survey. So here are some results from the preliminary model that I've created for yellow lent mussel across the range, with dark purple being unlikely present and then yellow being likely present. So we could see here some areas that we already kind of know are yellow lent mussel hotspots, like the Potomac and the Susquehanna drainages popping up in this model. I also tested alternative model approaches for this species, such as joint models and conditional joint models, and found them to not work out so well. So that's resulting in a whole other study that I'm conducting on why didn't these models work, which is in prep as well. Zooming in on a more local scale, here's a picture of my state where I live, the state of Maine. And in gray is the Penobscot River watershed, which drains about a third of the state. The blue line is the main stem of this really, really large river. And then finally, this zoomed in area is Marsh Island, where I currently work and reside. What's interesting about the Penobscot River is not only is it so big and so important to the people that live here, but two dams were recently pulled out here. The Great Works Dam, this white empty circle, and the VZ Dam, this white empty circle. And it's unknown how habitat or substrate and bed texture in these areas has changed prior to dam removal. We have some aerial images that we can see where the channel impounded by this dam may have been more steady. And now, since that dam has been removed, it's possible that habitat has changed. And it's become a more dynamic system. It's definitely become more difficult to vote on as people like me have to brave these now fast flowing areas. Thankfully, in order to assess whether benthic habitat has changed, we have side scan sonar data prior to when the dams were removed. So these benthic images of bed texture that we then redid and took more side scan sonar data in these areas to get at whether these patterns and texture have changed and to kind of assess, okay, is there still habitat for animals like yellow lent mussel in these areas prior to dam removal? What we found was that these roughness metrics that we generated via automated surface metric calculations haven't really changed more than could be expected just due to things like seasonality. So we can't say for sure that these dam removals change bed texture, which is actually a really good story to have as we continue to remove dams for restoration projects. Additionally, we're interested in if any of these automated roughness metrics could distinguish between habitat types. And what we found was while these roughness metrics couldn't get at what is a boulder, what is cobble from these sonar images, they could distinguish between coarse and fine substrate. So we're adding more sonar data into this model to see if these patterns hold true across sites. Finally, the last chapter of my dissertation was a big public outreach project. We, the University of Maine, collaborated interdepartmentally with the Wildlife Department and the Intermediate Art Department to create two concurrent classes to paint a freshwater ecology mural. We had people from Inland Fisheries and Wildlife, or our Maine state agency, come and talk about fish, mussels, bugs, etc., to these artists and ecologists who then work together to design and paint the Kunduskig Pump Station, which is a large building in downtown Bangor that is right on the waterfront. We have mussels here, that's an eastern elliptio poking out of the sand on the building, migratory fish, bugs, etc., all depicted on this building. We also created signs with species information and QR codes to our website that has really, really in-depth information about the life cycle of these animals, the importance of mussels, etc. So this was a really big project that we completed last spring. And I also published a guide on how scientists can interact and work with public artists and fisheries just this last year. So I would like to acknowledge the dozens of organizations that I worked with to collaborate and take ideas and share data with. I harassed all of you with dozens of emails, and I'm sorry, but I'm also very, very appreciative of you working with me and your patience. So thank you to all of the organizations here and more that I've worked with. That's all I have for you today. Feel free to email me with more questions if you're interested, if any of these topics really struck a chord with you. And again, I'm going to be talking more about species distribution modeling and that whole chapter on Friday. So you could expect to hear more from me if you're in the Yellow Lamp Mussel Working Group. So thank you all again. And I look forward to hearing from you.
Thank you. Thank you. All right. Any questions for Jill? All righty. We'll go on to our next presentation. Next up, we have Kim.
Hey, I'm Kim Morgan with Virginia Department of Wildlife Resources, and Kayla and I put together a conservation plan for Virginia's freshwater mussels with input from our mussel team. And let's see. Can everybody see that?
Yep, that looks good.
All right. So the purpose of the plan is to provide guidance on the conservation and recovery of freshwater mussels in Virginia. The introduction covers the ecology and life history, which includes anatomy, diet, reproduction, habitat requirements, importance of mussels, and their imperiled status. The declines of freshwater mussels nationwide, freshwater pearling, the button industry, the cultured pearl industry, dam construction, and also Virginia-specific declines, which covers chemical spills and mercury release. Threats to freshwater mussels, which includes habitat loss and fragmentation, host fish decline, poor water quality, invasive species, disease, and climate change. Our conservation efforts, dam removals and modifications, stream restorations, surveys and monitoring, propagation, augmentation of populations, and laws and regulations. The conservation strategy section has our conservation goal, the conservation actions taken or planned, surveys, propagation, host fish management, research needs, habitat protection and restoration, outreach, partnerships, and staffing needs for the hatcheries. And the third section is the species profiles. Each profile includes an introduction of the species, the distribution, any specific threats for that species, and then conservation efforts, a specific goal, and specific conservation actions for each species. And that looks like this is one for Varicose. It has a map which shows historic and current ranges, conservation efforts, and then specific actions that we would like to take for this species. And just some conservation success that we've had in Virginia. We've donated over 1 million mussels from our hatcheries to partners. We're doing ongoing long-term monitoring of wild and hatchery-raised mussels. 55 dams have been removed, which opened over 175 river miles for fish passage, and over 1 million mussels have been released in Virginia waters. That's it. I did not find any 16th place golf trophies, but I did find some nice river booty this past season.
And those look all sort of lucky. Thank you for sharing. Nightmare fuel and lucky. All right, does anybody have any questions for Kim? All right, last but not least for this section, we have Mike Walter. We maybe have Mike Walter. You here with us, Mike? Let me see if he popped back on. Alrighty, we might have lost him, but we are running a little ahead of schedule. Oh no, wait, he's here. He lives. Fantastic. You're still muted though. I lost you. Audio.
I took a self-appointed bathroom break. Sorry about that.
That's okay. At exactly the time I called on you, naturally.
No, of course. Let me get mine going.
Yeah, no worries. Take your time.
Okay.
Okay,
How's
we
that?
got your slide.
The whole thing?
Whole thing.
All right. Well, sorry about that. But first, I just wanted to say thank you for organizing this. I know I've fired off a couple emails today just from stuff we've talked about, so I'd call this a great success. I wanted to share a brief summary of over 2 decades of work that we've done here in North Carolina conserving the federally endangered tar river spiny mussel. So let's get introduced here. This is the tar spiny, kind of a nondescript mussel. It's endemic to the tar and Neuse basins here in North Carolina. It was first listed as federally endangered back in 1985, and it's a very small-bodied and long-lived mussel. So the longest individual in our database came in at a whopping 58 millimeters, and we suspect that they live somewhere between 20 and 40 years from our hatchery holdings and our findings in the wild there. But like Amber mentioned earlier, we haven't been able to do any age and growth work on this species just because of how rare and valuable those individual shells are and how destructive that process is. So hopefully that micro CT scanning will help give us some insight into their age. They prefer clean and consolidated sandy and pea gravel substrates like those that we find in the upper reaches of the tar and Neuse basins in North Carolina. And historically, they ranged throughout the tar and Neuse basins, so above and below the fall line, which is interesting. And they historically were found in Swift Creek, Fishing Creek, Little Fishing Creek, the Tar River proper, and Little River and Neuse River in the Neuse Basin. And just keep an eye on the scale of this map that we're looking at too. And now they've been constricted pretty severely to just a small corner of the upper tar basin, where the last individual was found in Fishing Creek was in 2016. We found one in Swift Creek of the tar basin in 2024. And that was the first individual that had been found there in 19 years. It was presumed extirpated from there. And then Little Fishing Creek continues to be our quote unquote stronghold for this species, where we're recovering 1 to 2 individuals per year. And those are brought into our hatchery and will spend the rest of their lives in our broodstock population up in Marion, North Carolina. But these surveys are extremely intensive and often take in excess of 150 person hours just to recover a single animal. And to date, there's only 11 tar river spiny mussels, true wild tar spinies that we know to exist, and they all live in our hatchery there in Marion. I'm sure there's more in the wild, but they all seem to be aging out in the wild. So it's a dire situation. But this trend has been ongoing since the early 2000s when biologists at North Carolina State and North Carolina Wildlife Resources Commission started noticing that they were recovering fewer and fewer tar spinies during their surveys. And when they were gravid, that those broods were largely unfertilized. And so they decided to start investigating some captive propagation strategies for this species. And this, again, was headed up in a partnership between North Carolina State University and the WRC here. So first they had to investigate some very basic life history traits that we all know. And they found that these are tachytictic brooders, and they're producing several broods per year between April and June. They package those broods as conglutinates. They're about 5 millimeters long. They're flat and ribbon-like. And most importantly, they identified 4 host fish species that were viable for captive propagation. Those being the white shiner, the pinewood shiner, which is a Tarnus endemic fish, the bluehead chub, and the satinfin shiner. So once they had those methodologies worked out, they got to work and they did a really good job of producing a lot of mussels. And to date, we've released over 40,000 individuals into now 18 different sites in the Tar Basin. And we've released nearly 3,000 individuals into 3 sites in the Neuse Basin. Each release, we use a variety of tagging strategies. So we do batch tagging where we're painting on black dot tags that identify entire year class cohorts. And this is a really time intensive and kind of fragile tagging method, but does seem to work for tagging a large group of animals very quickly. We use hull print tags like everybody does. These give alphanumeric IDs and seem to be very durable. We're finding animals that have been tagged for 10 or 12 years now that still have good, very legible tags on them. And we also do pit tags with these where this is glued onto the outside of the shell, either using dental cement or we've experimented with a couple different things recently. But these offer the best recapture possibility, but they are bulky. And with a small bodied mussel like this, there's some question of if it affects their natural movement or not. But they do seem to hold up pretty well. And then we also have a laser tagger in the Marion hatchery, which is a fast way to do both batch and unique ID tags. But we've found that after 8, 10 years in the river, that those etched portions of the shell are the most susceptible to erosion, and that those tags don't really hold up long term. So we do a lot less of that. So after release, we go back to all new established sites within 1 year of stocking and go check up on things there. We put the rest of our sites on a rotating monitoring schedule and try to visit all sites within 3 to 5 years or every 3 to 5 years. And 3 sites we've established as long-term quantitative monitoring sites in Little Fishing Creek. And there we do gridded quadrat surveys in 2 of those 3 reaches. And this gives us a lot of good information because we have a known stocking location for each unique ID tag, the individual, and that allows us to track things like movement and recapture rate, even habitat preferences because they were stocked into a variety of habitats within that reach. So this is some recapture data from the downstream-most site. It's worth noting that in the last couple of years between 2022 and 2025, there's a large tree that fell upstream of this and deposited a lot of fines and shifting sand on this site. So our recaptures have dropped off there, but we see that they're persisting in this site. This is the next upstream-most site. This is one that gets the gridded quadrat surveys. And the last major stocking event for this species was in 2018 there. And we see that those recaptures have pretty much plateaued. And that's a pretty good rate of recovery for us. We're happy with that. And then the third most upstream site is the gauge station. And it's also worth noting here in 2023, we went out targeting 120 individuals for a swabbing experiment. And so that's why that recapture was so low. But this is our most densely stocked site. It has individuals from every cohort that's ever been reared, and we're still recovering nearly 300 in a very short survey. So that's great. So the big takeaway there is that they are living once we're putting them in the river. So next we've been looking at annual growth for these animals. We've been seeing that they are showing signs of positive growth, albeit a very small fraction of a millimeter per year. But for a small-bodied animal, that's great progress for us. So again, they're surviving and they're growing. And a few interesting finds from this last I should have mentioned earlier, we do some really big, we call it the tar spiny roundup, where we pull in partners. It's about 20 or 25 people that come together and help us monitor these sites. We try to do that every other year. And 2025 was one of the years that we had that big monitoring effort. So one interesting find there was this individual green T626. And this animal was pit tagged before stocking and was put in the river at a length of 23.99 millimeters. And then it was recovered in each of the major sampling events after that. So in 2018, we recovered it and it was observed to be gravid. And this was the first sign that our stocked individuals were becoming gravid after release in the wild. So that was a major step forward in our understanding of this stocking success. And then we were able to observe it in 22 and 25 and show that it had put on some growth as well. So this individual shows the value of those pit tagged animals for us. we're really more likely to recover that animal and get that valuable long-term data. But more importantly, it gave us this indication that these animals are able to become gravid in the wild. So again, as we're evaluating the success of this stocking, we're seeing that they're surviving, they're growing, and they're even gathering the resources they need to become gravid and become sexually mature. So all positive signs there. A few other interesting finds from this last Tar River Spiny roundup was this individual T681. It was not pit tagged at all. It was first stocked in the river in 2015 with a length of 23.47 millimeters, and then went completely undetected for 10 years over 5 successive, very intensive monitoring surveys there before it was pulled out of the river for the first time in 2025, having grown 7.87 millimeters. And then that was true for 2 other individuals as well. So just a good indication of how bad we are at finding freshwater mussels in these visual and tactile surveys, and gives us a little bit of hope that maybe there's more out there than we're seeing in any given survey. But definitely interesting nonetheless that these could go 10 years completely unseen. So they're surviving, they're growing, they're becoming gravid. The next step in that life cycle is finding and infesting a target host fish. And so for us to understand if that was happening with these stock populations, we decided to go and collect those 4 known host fish species from directly over top of those stocked mussel beds. And so this was done at the gauge station site where I showed the recapture data from. And we collected pinewood shiners, white shiners, bluehead chubs, and then a slew of non-target host fish species, and just to understand what kind of host fish use was being done out there. All in all, after dissecting those gill filaments, we discovered what we thought were 10 glochidia and preserved those in 95% ethanol. 2 of those were visually identified as Tar River spiny mussel. They're a very large glochidia, they're spherical shaped, and it's pretty distinct. And then 1 was visually identified as the federally threatened Atlantic pigtoe. And we really wanted to be sure of what we were talking, or we wanted to be sure that we were correct in our identification of the Tar River spiny mussel on there. And so we turned to our genetics team, Heather Evans, and she addressed some of this work earlier. But we asked, are you able to do barcoding on a single grain of sand sized animal? And she confidently said yes. So we turned those samples over to her, and she was able to confirm that we did indeed have 2 Tar River spiny mussels, 1 on a pinewood shiner, that endemic fish in the Tar Neuse Basin, and then 1 on a white shiner. So that was a huge step forward in our understanding of these stocking successes that we've been doing. We confirmed that Atlantic pigtoe was on there on a white shiner, but we also had a lot of contamination of the sequences from the host fish tissue that was brought in with those samples. So they're exploring a number of things. They've tried host fish specific blocking primers in their amplification that had mixed results. They are next exploring the use of qPCR on the extracted DNA, and we're standing by for the results of that work still. So they're surviving, growing, becoming gravid, infesting host fish. Next, we should start to see some juveniles in the river. And indeed, in 2022, we had our first sign that things may be going in the right direction there when we found 1 untagged spiny mussel. This was in the gauge station reach, and it measured in at 19.7 millimeters. And this was smaller than any animal we had ever stocked there ourselves. So we felt pretty confident that this was the offspring of our hatchery produced individuals, and that was very exciting. The next major tar spiny roundup came in 2025, and we recovered 2 untagged spiny mussels then. And these were very different morphologically from the hatchery produced individuals because they had been born in the river, and their shell shape was very distinct. So we feel confident that those are the offspring of our hatchery produced individuals. And it's just a great story. It kind of comes full circle, and this is really what it's all about. After 2 decades of work, it's exciting to be able to share some of these results. So the next steps for this project, Heather mentioned that we have PBT panels, parentage-based tagging panels for this species and a few others. And so we're going to start asking questions of these untagged individuals and understanding how we can use those panels to understand hatchery contribution, wild contribution, and then just parentage in general there. We are exploring the use of eDNA to improve our broodstock collection for this species and make that more efficient. We're going to attempt to scan a river using that 300-meter known distance that the signal persists. And then when we find a positive signal for tar river spiny mussel, go in and heavily survey that area where we got the positive signal. And then we're going to continue to work on that extracted DNA from the host fish trials that we did earlier. But like I said, this is over 2 decades of work. And so there's many more people than who are on this slide who deserve credit for the successes we're seeing today. It's just our luck to be in the position to find these good results and be able to share it with y'all. But these people really deserve all the credit. So with that, I'll be happy to answer any questions.
Thank you so much, Mike. We'll wait to see if any questions pop up, if anybody's got some. Cool beans. I'm not seeing anything pop up. And so that wraps up our presentations for the afternoon, which is fantastic. The next thing I have on our schedule was just some dedicated time for discussion and synthesis and asking questions. It was a lot of material has been thrown at you really fast today, which is fantastic. So if anybody has any, I'm just going to leave it open for a couple minutes to chat about that. Maybe we can also throw in a bathroom break too. So if anybody wants to, feel free to start that discussion. Otherwise, I will move on if we don't hear from anybody at like, let's say 2:10. 2:10 sounds good. Okay, so I'll talk to y'all in a second. All right, welcome back everybody for the last little bit of the day. It seems that everybody seems to be good on discussion, so we'll just kind of cruise through that part unless anybody else pops up anything here. Maddie wants to talk about mussel food, so want to go for it, Maddie?
So this is, I was just looking back and Chantel kind of raised this back in my brain is what do mussels eat? And I mean, there's a couple of avenues that we can approach this through. I mean, both in the wild and propagation. And a lot of my experiments had to do with starving mussels, so taking away anything that would be good for them. But trying to figure out how much others have thought about the nutrition portion of this, and how then together we kind of combine each of these different angles for checking, you know, what is the optimum quantity quality that we are feeding, that we're seeing good results maybe in propagation facilities or in the wild, and then how we're tracking what happens if you take that quantity or quality of food away. Just not sure how much else others here have thought about that.
I promise your audio was on and they did hear you.
It's just one of those things that the foundational, you know, as long as you have your food, water, and shelter, then you're okay. And I think we have a pretty good idea about things that are in the water that take away our ability to be okay. And we're pretty good at knowing what kind of habitat these guys need. And then so, you know, where do we kind of, or who might be interested in doing a little bit more targeted, putting together what we do and don't know, and then splitting up the tasks to get a little bit better of a handle on it. Or is it even, I don't know, worth the thoughts, or are there bigger things that we need to be concerned about? Okay, I'll keep chewing on it.
Maybe what we could do is we'll throw it in the chat since I'll be sending the chat out. And we'll just put a call for anybody who's studying, what do you think, water quality out in the field, algae particulates, or what you're feeding in hatcheries?
Yeah, even a database of what we're seeing in the field or what other hatcheries are using that seems to be working for whatever species under whatever conditions. I know I've definitely seen some patterns, but that might only hold for some specific rivers. Others have seen, you know, what I think might be kind of counterintuitive is that they're feeding more on like, you know, fungal type things than even cyanobacteria or green algae. Not quite sure what all that means. Yeah, I'm not sure.
Yeah, this is Jay. I'd suggest reaching out to the folks at Virginia Tech.
Maddie, could you hear Jay?
Yeah, I got you. Thanks, Jay.
Thank you. Yeah, and Maddie, if you don't mind, since it seems to be a little bit fizzling, why don't you go ahead and post that in the chat, just what like specific action item the folks here can do to help you with that, and hopefully it'll peter in after the meeting.
Yeah, no problem.
All right. Any other questions you want to post to the group real quick? Okay, cool. I do have a quick one for everybody. After Maddie gets that up, let me share my slide. Mostly, I just want to start our wrap up here, say thank you all for coming so much. today. This turned out way better than I had expected. We had over 70 people in attendance. We had representation from Florida to Canada, which is amazing. So I did want to ask you real quick how you liked the meeting. More like, would you like this to do it again in the future? So let me share my screen as soon as I remember how to do that, even though I've done it 20 times today. All right, so I'm going to throw these two up in the chat real quick. Give a minute for, if you could throw those in there, that would be amazing. So I'm just wondering if y'all would be interested in a future virtual check-in like this. If you got value from today, I'm happy to organize it again. If it was cool as a one-time thing, that's totally fine too. My thought is maybe we could do it every other year, the opposite years of FMCS, if that would be helpful for people. I'm happy to continue just a virtual day like this so we can all keep in touch with each other. I also know that I miss people. I tried my best to get it out to as many people as I could, but if I missed folks here, I'm sorry, and I would love to include you on the next time. So if you could pass this on to them after the meeting here, I'll be sending out the everything from today. So the recording, the chat information, the contact sheet. So anybody who didn't get to join today, you can share that with them. And if they want to add their info to the contact sheet, that's what I'll use to get the word back out again if we decide to have this meeting again. So if anybody's got any thoughts on that, I'll leave this chat up and running for a couple minutes. And I think that's pretty much all I got for y'all. So thank you so much for coming, and good luck in all of your endeavors.
Thanks, Loretta. Good stuff. Appreciate it.
Yeah,
Thank you.